Good morning all and welcome you all to today's webinar, Frontiers in Biochemical Sciences, organized by Department of Studies in Biochemistry, Davangere University, in association with Society of Biochemical Sciences India, Davangere branch. Now I request Ms. Shalini KS to start today's webinar with invocation.
Okay, thank you, Gautami ma'am. Baro Krishnaya, Krishnaya, Baro Krishnaya, Baro Ninna Mukatoro, Riya, Baro Krishnaya. Krishnaya, Baro Krishnaya, Baro Ninna Mukatoro, Ninna Sariyaro, Jagadara Shilane, Baro Ninna Mukatoro, Ninna Sariyaro, Jagadara Shilane, Baro Krishnaya, Krishnaya, Baro Krishnaya. Anduke paadagavu kalanduke kirugeje, Anduke paadagavu kalanduke kirugeje, Dimdimi dimi dimi dimiranuta, Anduke paadagavu kalanduke kirugeje, Anduke paadagavu kalanduke kirugeje, Dimdimi dimi dimi dimiranuta, Pongulalanu duta baraiya, Pongulalanu duta baraiya, Baro Krishnaya, Krishnaya, Baro Krishnaya, Baro Ninna Mukatoro, Ninna Sariyaro, Jagadara Shilane, Baro Krishnaya. Thank you.
Thank you, Ms. Shalini. Now I request our department chairman, Professor Wadlapudi Kumar sir, to welcome the dignitaries as well as participants.
Very good morning, everyone. It's my privilege to welcome the dignitaries.
Good morning, yes.
And the resource persons, and as well as participants for this one-day webinar on Frontiers in Biochemical Sciences, being organized by our Department of Studies in Biochemistry, Davangere University, Davangere. And we have the branch of Davangere of Society of Biological Chemists India have been immense support for us in organizing these kind of events from our department. The Department of Biochemistry came into existence in 1993-94 academic year, and then Quebec University PG Center. Later, with the establishment of Davangere University, the department came into the fold of Davangere University since August 18, 2009. We in the department have been putting our best possible efforts to train our students and see that our department has been established one of the good research centers as far as Davangere University is concerned, with our contributions in the form of extramural grants, and as well as good number of publications. As a team, we have been working for the growth and development of the department and as well as our university. So today, we are organizing this Frontiers in Biochemical Sciences under the banner of Department of Biochemistry, Davangere University, and Society of Biological Chemists India, Davangere branch. So this event, it is mainly aimed for motivating the young generation and igniting the minds of PG and as well as UG students who are aspiring to achieve their future goals. And this is the event being organized with the involvement of young and dynamic scientific fraternity from different organizations of academia and as well as R&D industries and the research institutions. So we have Dr. PAVAN JUTUR from ICGB, International Center for Genetic Engineering and Biotechnology, JNU campus, New Delhi, who is going to give keynote lecture today on photosynthetic cell factories. And we have another young and dynamic scientist, Dr. SHIVAKUMAR KEERTHIKUMAR from Peter MacCallum Cancer Center, Melbourne. Both are our distinguished alumni. And we have Dr. VENKATA DK, a dynamic scientist and very active researcher from Central Institute of Medicinal and Aromatic Plants, CSR Institute from Bangalore branch. He's going to speak on role of lipids in signaling mechanism. And we have another distinguished personality, dynamic researcher who is associated with development of COVID vaccine from Biological E Limited, Hyderabad, Dr. VENKATESHWAR DM, who is going to speak on downstream processing of vaccine candidates. And our distinguished alumni, Dr. SOMASHEKARA, the co-founder of Olium Life Sciences, Chennai, is going to speak on his own experience so far with special reference to nutraceutical industry. He is heading nutraceutical industry over there at Chennai, and he is going to share his experiences with the students and motivate the students and as well as the participants from different parts of the country. And we have another manager from IPC Food Processing Division, Mr. VIJAYANANADA, our one distinguished alumnus, and he is going to speak on opportunities for biochemistry in industry.
Hello, Kumar, not.
Professor Kumar, you are not audible. Kumar?
Kumar is around.
Rangapur. Namaste, namaste. Bandinri na matartini. One idragi. Okay. Okay.
Sorry for the inconvenience. Please wait for a few minutes. Let Kumar sir will continue.
Link.
On behalf of Department of Biochemistry, I wholeheartedly welcome Vice Chancellor, Professor S. Alse sir, for this event of one-day webinar on frontiers in biochemistry. Sir, you are welcome for this inaugural session, sir.
Okay, thank you.
To welcome respected Registrar, Evaluation, Professor Anita H.S. Madam, for this inaugural event. Madam, on behalf of our Biochemistry Department, I welcome you for this Frontiers in Biochemistry National Webinar.
Thank you.
You are welcome. I also take this opportunity to welcome our resource person, Dr. PAVAN JUTUR from ICGB, New Delhi. I also welcome Dr. SHIVAKUMAR from Melbourne, Dr. VENKATA from CSIRC Bangalore, Dr. VENKATESHWAR from Biological Limited Hyderabad, Dr. SOMASHEKARA from Orient Life Sciences, Chennai, and Mr. VIJAYANANADA from ITC Limited, Bangalore. I also welcome my own colleagues in the department, senior Dr. Panjita, Ms. Sharani, and dear students, and as well as all the participants. And I once again welcome you all, and I thank for the opportunity given to me to welcome you all. Thank you, one and all.
Thank you, sir. Now I request today's guest of honor, our respected Registrar Evaluation, Professor Anita H.S. Ma'am, to speak few words about today's webinar.
Okay. Good morning to all of you. Honorable Vice Chancellor S.V. Halse sir, Registrar, Finance Officer, and all other specially invited speakers on online platform, research scholars, students, faculty members, and syndicate members who have joined this program. We are very happy that our university departments have been conducting so many webinars. Every day, one or the other webinar will be there. This is also one such webinar which is being conducted by Department of Biochemistry in association with Society of Biological Chemists, Davangere branch. And the title of the webinar itself is very catchy, Frontiers in Biochemical Sciences. And one more interesting thing which we could learn, that all the speakers who have been invited spread over the globe. One is speaking from abroad. The others who have joined this online webinar, who, very interestingly, many of them are alumni of our department, Biochemistry department. We are very proud that they have extended their support in conducting this webinar. So on behalf of our university, on my own behalf, I extend a very warm welcome to all of them to our university, and I congratulate Professor Kumar and his big team for having initiated this particular webinar. One day full tight packed webinar with very interesting topics. So I congratulate the whole team for having taken up this, and I thank for the opportunity given to me to speak few words. Thank you.
Thank you, ma'am. Now I request our Honorable Vice Chancellor, Professor Sharanappa V. Halse, sir, who is presiding today's webinar, kindly to speak presidential words.
Good morning to all. Davangere University, Department of Studies in Biochemistry, and Society of Biological Chemists, Davangere branch. Today's speakers are PAVAN JUTUR, Group Leaders of ICEB, New Delhi, and SHIVAKUMAR, he is from Australia, and VENKATESHWAR, Principal Senior Scientist, CSIR, Camp Bangalore, Venkateshwar Reddy, M. Managing Director, R&D, Biological Limited, Hyderabad, SOMASHEKARA N., Co-founder and CSO, Olan Life Science Limited, Chennai, Mr. VIJAYANANADA, Managing Food Division, ITC Limited, the Bengaluru, all syndicate members and academic council members, Dean of the Science Faculty, Registrar of this university, Register Evaluation, Finance Officer, and invitees, those who have joined for this webinar, Professor Odalpudi Kumar, Dean, Faculty of Science, Chairman of the Department, Professor Gopal Advirao, Professor Pramod, Dr. Satish Kumar, Dr. Purnima, Dr. Rajshekhar, Pratap GK, Miss Ranjita, Miss Hulina Salini, researchers, student of the department, all participants for this webinar. Topic chosen for this webinar are very important for the faculties and researchers for this webinar. And Department of Studies of Biochemistry has been actively involved in imparting knowledge of biochemical sciences on studies, making the significant contribution in the academics and research activities in the university through extemporal grants. The department organized the event of this kind of through physical gathering and virtual mode almost every year. I congratulate Department of Studies in Biochemistry for our Davangere University and conveying my best wishes to Professor Gopal M. Advirao, Professor Kumar, faculty members, research scholars, and the students of the department for organizing this webinar and inviting online the young and dynamic scientist personalities from the academic, R&D organizations and industries to enlighten the students about the latest development in the recent trend in the field of biochemical sciences. And for this, this COVID-19 pandemic, so as to motivate the students and research scholars to plan their future career, I wish the one-day webinar on 20 years in biochemical sciences a grand success. Let many more events for this kind are organized by the department in the days to come. I once again congratulate the team of the biochemistry department. I thank for this opportunity. Thank you one and all.
Thank you, sir. Now I request Dr. Santosh Kumar, sir, to deliver a vote of thanks to thank all the dignitaries.
Good morning to all. It's my immense pleasure to present a vote of thanks on behalf of myself and Department of Studies of Biochemistry. First of all, my sincere thanks to our Honorable Vice Chancellor, Sir, Professor S.V. Alsay sir, for gracing this occasion. I want to extend my sincere thanks to Register Department of Register Davangere University, Professor Gayatri Devaraj Madam, and Register Evaluation, Professor Anita H.S. Madam, for their support and encouragement. And I extend my thanks to Professor V. Kumar sir, Chairman, Department of Biochemistry, and Dean Science and Technology for their leading support and inspiration to every point of time. And also thanks to Professor Gopal M. Advirao sir for their support and cooperation for conducting these webinars. On behalf of Department of Biochemistry and our university, I am very thankful to all the speakers, resource persons, and thanks to all teaching staff, teaching faculty, research students, non-teaching staff, and other department faculty members and students. I'm thankful to all all the participants for joining this one-day webinar and for joining for this inaugural session. Thank you for giving this opportunity. Thank you, one and all.
Thank you, sir. Now it's the time to start our scientific sessions by various resource persons. I kindly request all the participants to get maximum benefit of all the sessions. If you have any questions or queries regarding the talks, please post your questions in the chat box in Zoom platform and in comment section in YouTube. Let us start our keynote, our first session. I request Mr. Ashekar sir to introduce our first session speaker, that is Dr. PANNAGA PAVAN JUTUR, Group Leader, ICGEB, New Delhi, on the topic Photosynthetic Cell Factories, the fourth dimensions in industrial biotechnology.
It's my privilege to introduce Dr. PANNAGA PAVAN JUTUR for one-day webinar on Frontiers in Biochemical Sciences. Dr. PANNAGA PAVAN Kumar JUTUR is a group leader, Omics of Algae, Industrial Biotechnology, International Center for Genetic Engineering and Biotechnology, New Delhi. He obtained his postgraduate degree in biochemistry from Kumpi University, that is Department of Biochemistry, Kumpi University PG Center, Davangere, and MPhil PhD from Pondicherry University, Pondicherry, India, in the area of biochemistry and molecular biology. Later continued his research as postdoctoral research associate at University of Nebraska, USA till 2012. Dr. Pavan JUTUR joined ICGEB in 2012 and his primary focus of research is understanding the dynamics of microalgal systems through integrative multi-omics approach in coherence with reconstructed genome scale metabolic models with well-defined functional pathways that will elucidate an effective strategy for converting lighter carbon source into biomass, biofuels and biorenewables for sustainable futuristic solutions. Dr. PANNAGA PAVAN research group's findings will provide important breakthrough on central metabolism in microalgae, which are required for biotechnological improvement of next generation biofuel production. Dr. PANNAGA PAVAN has published significant number of research papers in high impact internationally reputed journals. Today, Dr. PANNAGA PAVAN is amidst us online to deliver keynote lecture on photosynthetic cell factories, the fourth dimensions in industrial biotechnology. Sir, I request you kindly to lead the session and nurture our students with your valuable contributions. Thank you, sir. Thank you, Anand.
Thank you very much for your kind introduction, and I am very happy. Virtually, I have been in Davangere after 21 years, so I finished my MSc. Some of my teachers are still there: Dr. Gopal, Dr. Kumar, Dr. Purnima. I hope they remember me. So definitely, so has been a long time, and I'm very happy and thank university for inviting us to share our experience, what we are doing at different institutes, and it's a very great event to inspire the students from nowhere to somewhere. So I'm very happy to be a speaker today, and unfortunately, we cannot meet in person, but due to COVID-19, but definitely I will be happy to visit Davangere sometime when everything comes back to normal. And so today, today is a great occasion for us to talk about different areas. So can you please give me the sharing screen option, please? So please bear with us. There is some technical issue.
Sir, you can share now the slides. Pawan, sir,
Okay.
you can share.
Yeah, but it is enabled. It is showing enabled. Can you make me co-host or something?
No, sir, you can share now. It's opening, sir.
Yeah, it's coming. It's coming. Okay, just hold on. Yeah. Okay. Yes. Perfect.
Thank you, sir.
I hope the slides are visible now.
Yes, sir. It's visible.
Oh, okay. Just bear with me for a second. Okay. Thank you. Okay. Thank you very much again for inviting respected Vice Chancellor of Davangere University, Register, and all the faculty members of Biochemistry. It's my pleasure and honor to present our scientific research, what is happening at ICGP. So I want to introduce myself again. My name is PAVAN JUTUR. I'm Group Leader of Omics of Algae Group, Industrial Biotechnology at ICGP. So basically, we are working on third generation and fourth generation biofuels, because biofuels are one of the hot area of research throughout the world. So today I'm going to share how we use multi-omics approach to understand the basic biology. So being a biochemist, we are always interested to look into the cell, what kind of pathways are regulating, what is happening within the cell when a particular treatment is given, and all those things are very important. So to have the gist of everything, so I'm going to present few topics of a few of my scholars today, which will give an overall view of what we work at ICGP. So today's topic is photosynthetic cell factories. Why? Because we work mainly on algae, which are microalgae. So because microalgae are the one source which can take up the solar energy and convert into multiple products. So that's why they are photosynthetic cell factories which can be able to generate. And the fourth dimension in biotechnology is that the future of biotechnology lies in one of the main threshold of these cell factories. Why it is been, after the talk you'll understand slowly. So to start with, so this the picture in the background, what you can see is the Rubisco protein, one of the largest protein on the earth, especially in plant family, but microalgae are also known for having Rubisco. And this is the structure we have first time prepared through a computational approach of nanochloropsis. So this is a species which is very important in production of omega-3 fatty acids. So the fish what we eat contains of lot of omega-3 fatty acids. Basically, the omega-3 fatty acids in the fish are from the microalgae, consumption of microalgae, which produce omega-3 fatty acids. So this is one of the area where we are working largely upon, and I will be highlighting some of the research aspects in it. To go further, so I want to just give a few slides on what is ICGB. So ICGB is International Center for Genetic Engineering and Biotechnology. So we are situated, presently I'm situated here in Delhi, and our main headquarters is in Italy, and we have one more component in South Africa. So there are three components, and internationally we are located in different locations where we are promoting research in terms of field of life sciences and biotechnology. So we don't give any undergraduate or graduate program, but we are only completely a research organization who basically work on training and technology transfer in the fields of different life sciences and biotechnology for promoting sustainable global development. So these are the few member countries which are associated with ICGP. They are 67 member states and 20 signatory countries. And as I told, there are 3 host components of ICGP located in India, South Africa, and Italy. And all the other member countries are also in network of with different biological experts having a same goal of working on different biotherapeutics, diagnostic, agriculture sciences, and biofuels. So we at our ICGB, there are different centers internationally also. These are the core macro areas of research we work upon, that is infectious disease, non-communicable diseases, medical biotechnology, industrial biotechnology, and plant biology and biotechnology. So these are the core areas where we work upon in different areas. Among that, I am in industrial biotechnology, where we work on biofuels and other aspects. So ICGB not only gives fellowship programs for after finishing the post-graduation, they have a number of scientific seminars and courses which can be organized together with ICGB, with other universities nationally or internationally. And also to young faculty who are interested to establish their research can apply for the research grants with other components internationally, so that they can get a small seed money around 75,000 euros for a couple of years, where they can invest their research on in collaboration with ICGB scientists. So these are few opportunities and what are the socializing activities what we do at ICGB to in brief. And as I said, like we work on different diverse areas of biotechnology and genetic and engineering, and basically we work on basic sciences to understand in different systems, and also nowadays because any basic science which cannot be translated into industrial level is required. That's why translational research is also being included in some of the few projects where we are involved. With that content, an introduction to ICGB, what we do at ICGB. So I would like to just share a small content today, what I'm going to discuss with you guys. And if you have any questions, you can always email to me, and or you can put in the chat box. I'll try to answer that. So anytime you have, you let me know when I should stop, because I know that we have limited time, and I'll try to present whatever I could try to focus upon. So today I'm going to discuss upon what are biofuel generations, because especially for master students who are interested to nurture their career in terms of research industry, because this is the area which is going to flourish. And yesterday also you can see that there is a huge project on deep ocean studies which has been approved by the government, which is involved with, again, indirectly or directly with energy generation. So it is very important to know what is the biofuel generations upon, why do we need biofuels especially, and then what kind of feedstocks in terms of algae are present to make biofuels and other co-value products. And there is always a big question that we hype a lot of things about biofuels and whether it is possible or not. So this road to biofuels explains is possible, and in US they have shown that it is workable solution. And in India also we are going to have great results in terms of improving upon this to the next level of industry. So omics of algae, B3, what we call as biofuels, biomass, and byproducts is one of the core area what we are working upon, what we call B3 algae. So I'll be talking more about our research there. And the fourth dimension is what is the futuristic approach, what the students have to think for the research, and how, if they're joining somewhere or they want to join somewhere, what is the scope of research they have to look for. All these things comes in the fourth dimension to conclude with. So we know that fossil fuels are going to be diminished by 2050, and we need an alternative source for the fossil fuels, especially the conventional fuels. So it is estimated that already 93 million barrels per day are required in terms of fossil fuels with different activities throughout the world. But by 2030, it is estimated that this requirement may diminish, may come down due to the diminishing fossil fuel resources to 29%. by 50-50, almost it goes to zero or nothing. So there should be some kind of a alternative, and the demand by 2030 will be increasing to approximately 110 million barrels per day, which is huge in terms of what kind of alternative resources can then. So there are different undeveloped or unidentified sources which lie nearly 57%, where you can target these sources to be kind of one of the things what we are going to talk about is third generation biofuel, which is an alternative for fossil fuels, especially for biofuels like biodiesel, jet fuels, and other high value products. So this is a very important slide in order to understand what are the biofuel generations. So basically it comes under three sections: raw material selection, what kind of raw material should you take in order to select your type of biomaterial you produce. And the second one is technology of transformation, where it depends on how you take the raw material and convert back into the product of interest, like biodiesel or anything. And finally, the product selection, it may be high value product, biofuels, biojet, bioethanol. All these all comes under the products like bioenergy, biomaterials. All this comes under the product selection. So basically, if you see this threshold where raw material, technology, and product selection is one area, the other one is based on the what kind of feed you take. It comes under first generation, second generation. The first generation is sugarcane, rice, wheat. It belongs to an example of food crops, and especially the first generation is no more possible because it gives a conflict of interest between the food versus energy issue. So people started with first generation biofuels, but they know that food is equally important in terms of population consensus. So we need to have some second order generation, which is especially with which is lignocellulosic biowaste like cellulose bagasse or forest residues, grass, cell biomass for fermentation. All this comes under second generation. And basically, second generation is a fermentation biology where you can produce bioethanol. And by 2025, our respectable Prime Minister has made a mandate that 10% blending of bio alcohol or ethanol should be there with the fossil fuel. So it is one of the main important relevant target where we are looking, and in our center we work on especially enzyme technology and fermentation, colleagues of mine. And third generation is especially from algae, what I'm going to talk about more on that. So we have algae as a third generation biofuel, and especially because algae can produce naturally lipids or fatty acids, which are very important. This can be converted into biodiesel, which are the precursors, biodiesel molecule, biodiesel or biojet, and also algae is the main. The fourth generation is, that environment has a lot of CO2 pollution and the environmental hazard. So how to fix this CO2, how to capture this CO2, which is carbon sequestration, either by plants or algae, converting them to useful products like biodiesel or bioenergy or whatever biochemicals it can be, is the fourth generation non-edible crops. So that's how the raw material is selection. Once the raw material is selected, you go for different treatments like physical, thermal, thermochemical, chemical, and biochemical in order to produce a product of interest. So all these things is the one which adds to the cost, which is capex and opex, which is capital cost versus operation cost, which industry talks about. So today I'm not going to focus on much on technology and all because it is the main thing, but still this is the cost factor which involves, because you have to convert the raw material, extract the material to the product. So this is where the cost is involved. Finally, products, that you can use as biodiesel, bioethanol, you can use as petroleum sources, jet fuels, and biochemicals and food products. Altogether, this is where different biofuel generations come upon and lays a key important relevance in terms of how you can select the thing. And these are the core areas. Also, a number of oil companies are establishing these biofuel industries in coming or distilleries, what we call, for bioethanol production is the priority at now, but biodiesel and algal research will be the next generation preference. So as I said, why we need biofuels? Fossil fuels are limited, global energy demands increasing, and CO2 always released from these fossil fuel oxidation causes climate change. And now we have that climate change agenda to reduce the pollution contaminants in the air. India is also one of the very highly involved energy partner in order to reduce these CO2 into the atmosphere. Lipid petroleum transport fuels are very hard to replace with other renewable sources like wind, solar, and hydro. They have their own advantages, but we need liquid petroleum transportation fuels, the majority of that. Last but not least, biofuels are carbon neutral. That means the net carbon which is being sent back into the atmosphere or into the environment is negative or always zero. So that means that you are not contaminating your environment. That's why biofuels are more important and the main source as an alternative for the diminishing fossil fuels. So as I said, like there is a classification of algae. Algae can be explored. This is where the many species of algae are present in the world, and India being having a huge coastal line, coastal water resources. As I said, like Deep Ocean project, which has been inaugurated yesterday, will be the one of the niche where you can have more and more algae, which can be found out from the natural resources. One of the main important three classifications of algae, which you might have studied in our master's and graduation program, especially cyanobacteria. Cyanobacteria are single-celled organisms, which is one of the popular cyanobacteria everybody knows is spirulina. It's a protein-rich cyanobacteria, and it can be edible. And already you have spirulina in market, which has a huge relevance in terms of protein, single-cell protein. So these are different types of, I'm not going to talk about the physiology of cyanobacteria or algae, because I have to show you a nutshell, especially for postgraduate and graduate students. These are different types of cyanobacteria. These are microalgae, especially my group works upon. So we have red algae, we have green algae, we have brown algae, depending on what kind of products we are expecting. One of the very important microalgae, what I'm going to discuss today, is one of the nanofluoropsis or microfluoropsis, which can produce omega-3 fatty acids. Okay, and also the other astaxanthin, lutein. These are nutraceutical and pharmaceutical products, which are very important in terms of produced by these algae. One important model system which is extensively studied in microalgae is Chlamydomonas. Everybody knows about Chlamydomonas, and is a very important organism. So these are the few examples I'm showing here, what kind of microalgae we work upon, and followed by macroalgae, which is seaweeds. So if you are very fond of sushi food, you know seaweeds are the part of sushi food. The thin seaweed, what is around the coating of every sushi has the seaweed, and which is very high nutritious carbohydrate source. And these are the different types of seaweeds. And this area of macroalgae is still unexplored, and this is a fantastic biomass resource, especially for fourth generation, because it has a huge advantage and it produces huge amount of carbohydrate or sugars, which can be converted into bioethanol. There is a high scope of developed research in this area, and macroalgae are also present in a high biodiversity within India and other sources. Coming to why algae, as I said, algae has huge advantage because they can produce high oil productivity per acre than any land plants or crops. Use of non-arable land, especially they can be used in variety of water sources like seawater, estuaries, marine sources, because fresh water is very important for livelihood of human beings. So it can be grown in wastewater treatment of rivers or plants or oceans or whatever it is very important, and it's sequestered by or treated by algae. It's a huge hot area of research which is going on. As I said, mitigation of greenhouse gas emissions by algae. And finally, it not only produces biofuels, but also produces valuable co-products. It is very important not to, so In either way, you can engineer algae to produce your products, and GMO products can be grown in closed bioreactors to produce product of interest. And you can also media engineer. That means you can modify the natural conditions such that it can produce the same metabolite or product at a higher rate. So with this few this thing, and there are challenges why it is not feasible and all, large scale production, harvesting, all these things bring to the high cost. Presently, the algal fuels are facing with. People are working, and we are confident that we can bring down the cost. That's why the example, whether the algal biofuels are feasible or not, yes, definitely. This roadmap is the best example, which is developed by a which is large consortium, and at NA, which is known as NAB, which is which shown that the algae can be cultivated and harvested, and it can be derived into fuel and fuel derivatives along with other high value co-products, which will bring down the cost. Basically, the roadmap shows that algal biofuels are sustainable and very relevant in terms of futuristic approach, where we need alternative sources, alternative sources of fossil fuels is sustainable and feasible and also renewable. So this is a kind of a roadmap, and you can get this report online, because it's very important to understand whether it is feasible, why we are working on that. So this is a small video I want to show in relevance with algae, this thing. So just bear with me. Okay, sorry. Okay. So that was a small ad from ExxonMobil. Says that algae is the best source, and even I'm small, I can do big things. And it's very inspiring to watch that video, which is available in YouTube. So it's a production of ExxonMobil. So coming to our group's research, I'm going to introduce you with a new term called omics. You know, genomics, proteomics, transcriptomics. So this is a very versatile analytical tool which is being used everywhere nowadays. Without that, basic sciences is impossible. So there are different kind of omics is a collective characterization and the quantification of biological molecules within a cell, and that can be translated into a structure, function, and dynamics of an organism. Basically, it will visualize what is happening within the cell and tell you what is exactly happening, so that you can modify and manipulate according to the pathways and what you are interested in. There are different types of omics, and this is a famous project of NASA, which they're working on omics of astronauts by giving different supplements of proteins, DNA and all, and see how they are doing with the metabolism process, employing the omics perspective, because when they are abroad, when they are in space, how these astronauts can come up with different metabolism is the study of importance using omics. So omics is a translational field which is having a relevance in different areas. So especially this is one of the best examples. So at ICGB, what we do is we focus on multi-omics approach. So people work on single platform kind of omics, focus on one or two molecules, or single omics like transcriptomics, genomics, to understand the behavior of the physiology of a cell. But nowadays, because of technology advancement, we are employing a approach called multi-omics approach, where you can have different layers of from genomics, transcriptomics, proteomics, fluxomics, and metabolomics together understand a whole scenario of what is happening within the cell. So the omics revolution is an integrated comprehensive omics approach, combining all these for the advanced understanding of molecular mechanisms of the cell. So just like forward omics and reverse omics, we employ both the methods. So for example, genome information is present. You can start with DNA and go with until metabolites and understand what is happening within the cell. Whereas if you don't have any information like transcriptomics or genomics, you can do the metabolomics and map down to the proteomic onto the pathways and see what is happening, and then indirectly going to the genomics. So reverse omics is the one which I'm going to discuss today, which is the latest field of research, what we are trying to understand through non-sequenced microalgae, which are native and also which are universal, where they are present and then can be grown in any condition. So these kind of approaches. So basically, we are understanding what is happening within the cell in that context. So basically, as I said, the omics of algae works on three main components, B3, which is one is the biomass, how to produce or enhance the biomass. Second one is biofuels, especially what I was talking about. Third one is byproducts like astaxanthin, omega-3 fatty acids, tocopherols. These are very industrially relevant things. So the first topic is biomass. The subtopic is biomass. And the Mukul, my PhD student, final year, I'm going to present his work in microchlorosis gadiacana. So basically, the rationale or hypothesis for his study is he's supplementing carbon. So he's supplementing with additional carbon like CO2 and employing multi-omics approach to understand what is happening to carbon fixation mechanism, because microalgae or plants or macroalgae has carbon concentrating mechanisms in their systems, which is evolutionary, and how to enhance the cell biomass through that by engineering the regulatory network. This is his central hypothesis. So what we have done is we have taken normal air conditions where carbon dioxide is very less or very minimal, 0.03%, which is called VLC condition, and HC, which is in LO, is high carbon condition, where you change different concentrations of CO2 from 3%, 6%, 12%. So microalgae reports are capable of equal sequestering up to 18% CO2 directly into different products. So this slide basically says that at high carbon concentration, that especially at 3%, the biomass or proteins or carbohydrates gets enhanced in terms of this microalgae and also lipids. So this whole slide dictates the biochemical process, how the physiology. So basically, we see the physiology, how is the supplementation of carbon is enrolling. So if you are interested, you can go through this publication. So basically, what we have done is a feedback or reverse omics approach. So we started with metabolomics. So we took the samples and we have removed the protein, and the metabolites were either derivatized or taken into extracted into a suitable media, and we run in a orbitrap or a GC or LCMS to see how many metabolites are coming, and that data is automatically, or we have analyzed through different platforms like Metabolomics, Protein Discover for proteomics. All these things are done in-house, starting from extraction to the data analysis. So it is very important. So our group works not only in the wet lab experiments, but also bioinformatics is the strongest area. So when we done metabolomics, there are like different kinds of byproducts, metabolites, which were upregulated and downregulated, especially this comes of fatty acids, sugars, amino acids, organic acids, and others. And this is how they're upregulated and downregulated in course of time with low carbon and high carbon. This is how we try to understand within the cells, and finally the hypothesis is established in such a way that when carbon is low, the carbon is fixed through carbon cycle through riboscope, and it produces starch molecules or carbohydrates along with which is enriched by other stress molecules like trehalose and all. So basically, at low carbon, when carbon is getting fixed as carbohydrate rich molecules, whereas at high carbon, the same process happens, but it also enhances high value products like tocopherol, lipids, and other amino acids and glucoses. That's how we can enhance the lipids in the presence of carbon flux in high carbon conditions. So in order to understand this, the same thing we have done the proteomics to overlay what is happening with the metabolites, and we have found that there are a number of proteins which are upregulated during VLC and HC conditions within this. So basically, we do in time course because we have to know what is happening. So if you know that this microalgae doubling time is 24 hours or 48 hours, depending on that, we dictate a time course of 0 to 1 to 10 days or 20 days, depending on what product you are interested in. So this is a time course map for 24 hours because carbon concentrating mechanism in microalgae is very important in initial 24 hours. And so in order to understand what is happening based on our metabolome data, we understood the proteome. This is a nutshell what happens with the proteome analysis. You can see all the green components are with VLC conditions, and the blue which are labeled are with high carbon conditions. Basically, what we assume that the proteins which are upregulated in green are shown in green are the metabolic pathways which are upregulating during the low carbon concentration. And the light for 16 hours is shown as a light with white background and gray background is dark. So usually you do experiments in a diurnal cycle, either light and dark cycles or full light cycle, full dark cycle, the different experiment, but this experiment is 16:8, where at the dark, the lipid or photosynthesis or other biosynthesis happens in terms of high carbon, and the Rubisco fixes this carbon and that goes to extracellular lipid molecule. So in order to understand how this Rubisco is fixing, we have done some cell dynamics, and especially in this microalgae, because Microfluoropsis gadiatana Rubisco is not structural available. So we have taken another route of amino acids where we have taken these amino acids and try to compare with the closest to model system, which is having 52% identity, and with Thalassovia it is 76%. These are all microalgae which I'm talking about similarity, and we have constructed this beautiful Rubisco which has 8 subunits, and it belongs to Rubisco 1, C, and D. So Rubisco A, B, C, D, different kinds of Rubisco are there which can fix up, and especially in Microfluoropsis gadiatana we have 1 CD, and it has a small unit and large unit, which is the larger unit is the catalysis unit. The small unit is not involved in catalysis, but still it is required for combined protein. So we are working on the dynamics, how RUBP gets fixed contacts with this Rubisco molecule. This is the small video which shows, I'll just show the video, switch on the video. Yeah. So this dotted line shows the interactions. This dotted line will show the interactions between different RUBP, which is the first interaction molecule, 5-carbon molecule, and with the Rubisco, and how the CO2 comes combines with RUBP to form 6-carbon molecule, which you read in your biochemistry. So Rubisco, how it fixes carbon dioxide in form of sugar, 6 molecules. So this is where the interaction shows, and this is ongoing work which we are trying to work upon, how CO2, O2 competes with each other when with the Rubisco, and we are trying to see the dynamics of how it can work. In nutshell, this microalgae, Microfluoropsis gadiatana, can produce a lot of lipids and especially omega-3 fatty acids. And also we want to understand how the carbon is getting fixed up in this due course of time. This is on biomass natural, and so this is on biofuels where my postdoc, so he's my first student who has finished the PhD, and he is going to Sweden now for his postdoc, and he works on biofuels, and this is what the work on biofuels they are going to. So he worked on nitrogen stress, and they have shown that how lipid accumulates within the within this biofuel. So this is one more. So when you remove the nutrient stress, the basic biology here is when you remove the nitrogen, phosphorus, and sulfur, these are the 3 main components of the algae in growth media. So if you try to remove this nitrogen, which is very important part of our protein you know that nitrogen, protein contains full of nitrogen. So now it requires nitrogen. So if you remove nitrogen, for all the cellular mechanisms get affected, including photosynthesis and beta oxidation. But the carbon should not be wasted, so it gets accumulated in the form of triglycerides. This is an example of microscopy where you can see within the green microalgae the brownish or yellowish large oil globules, which is nothing but lipids, which are the biofuel precursors, which are I'm talking about lipids and all. So when you replete back, this lipids disappear and the cells become normal. So this is where one of the area which is still unexplored, and you can see that how you can take up the cells and replenish back once you extract the lipid, and you can get it done. So this is one of the. So we call in stress biology here, where you remove the nitrogen, phosphorus, and sulfur. You see this basically this slide tells you like within in a time course the lipid gets accumulated within 10 days. Huge amount of lipid gets accumulated as I shown in last time. So nitrogen is one of the primary stress mechanisms where you can accumulate large number of neutral lipids. Neutral lipids are the ones which are required for biodiesel, especially because of their composition. So again, we went with targeted reverse omics approach. So as I said, this is one of the this is the approach what we go upon, because the genomics of that was not published that time. So we were the first to. This is especially in Parachlorella castelli. This is one of the Indian isolate which is isolated from Bay of Bengal. So and that's what this is a best example how you can explore the Indian isolates, because we need Indian isolates in order to have an industry in India, and because the climate plays a major role in microalgae growth and other stuff. So this is the metabolome data, which I will not tell in detail. As I explained it, the different metabolites which are upregulated and downregulated with the nitrogen stress, and this is the overall what we see within the cell when the nitrogen is upregulated. This N minus is the one which upregulates a particular metabolite like citrate, alpha glucoglutaric acid, and that is converted into pyruvate, and that pyruvate gets indirectly fixed into fatty acids or tags, which are called triacyl glycerol molecules. These are the red lines which you see are the ones which are upregulated, and the blue lines are the ones which are downregulated in terms of nitrogen metabolism. Thus, we try to understand or visualize how it is happening within the cell, and we give a particular condition, and we need a condition where we can produce a lipid continuously without the compromising the growth. So in order to understand, we done the proteomics of that, and this is the proteomics profile of the Parachlorella castelli, where we obtained 166 significant proteins with respect to nitrogen starvation. So then we went for transcriptomics and genomics to understand and overlay the proteomics and metabolomics data in order to understand what is happening within the cell. I'm not telling about the details. If you're interested, we can always discuss in detail, because each technique itself is a 1-hour lecture, and it's a very interesting science where we do transcriptomics through Illumina, and we have different kinds of transcripts which are upregulated and downregulated. You can see the blue lines which are transcriptomic and genomics, and the pink one is the overall metabolic pathways which are seen. In the insert, you can see the red ones which are upregulated genes in terms of nitrogen starvation, which I am discussing about, and the blue ones are the downregulated. That means they don't have a role in nitrogen starvation. The red ones are the ones which are upregulated. That means that each of these metabolic pathways which are upregulating the proteins, which one overall you can see from the protein metabolite and all these things, we integrated the data, and this looks like this. And what is happening within the cell is through glycolysis, it gets fixed through the half TCA cycle, which is very popular in terms of stress, where it tries to save its energy and converts into acid pyruvate, and the pyruvate goes into back into ER, where it converts into fatty acids. That is nothing but triacyl glycerol. So the red line, that red arrows indicate that upregulation of these pathways are happening, and blue downregulation. And these are the integration of transcriptomics, proteomics, and metabolomics in a nutshell to understand what happens to the lipid pathway when we subject to starvation. So this is a very complicated figure, and in a simple nutshell, we have overlaid. This is first time we are generating a hybrid genome assembly in India in ICGB, and top to bottom work is being done at ICGB, including the analysis part. And we have seen, we only except for sequencing, we are outsourced to Illumina and Nanopore to get the data done. But other than that, everything is done at ICGB. And this pink pathways which are highlighted are the ones which are connected to lipid biosynthesis, and the other pathways are being also shown. So when we integrate everything, this is what happens within the cell. The different genomic aspects, proteomic, everything are overlaid, and we try to understand each pathway in different terms of what is happening with different pathways. And the yellow indicates the metabolites which are upregulated and downregulated. The red indicates the transcriptome, green indicates the genome. All these things integrate together, give a concise picture how within the cells the metabolic pathways are interconnected and what are the targets to manipulate. So that is how we try to manipulate. We try to understand through multi-omics approach, and that can be included in the systems biology model, where you can understand before doing the experiment. You can put in the model, mimic in the model, and see whether you are able to accumulate particular pathway. And we are also, as I said, we are reconstructing the pathways to understand through these omics what is happening within the pathway, what kind of pathways are upregulating, what kind of genes and transcripts. Thus, you can dictate these microalgal or photosynthetic cell factories, what kind of product you can produce in terms of understanding the biology, basic biology of the cell. Finally, I'm going to discuss about Ikra Mariam's, my final year PhD student, who works on bioproducts, especially omega-3 fatty acids. So just give me 5 minutes, I will be finishing this. So basically, she also uses different carbon substrates because biomass is the main constraint, and she has to produce omega-3 fatty acids. And you know, the omega-3 fatty acids are having a huge market in terms of by 2025, it has almost 3,450 million by market in world. So there are different companies which are already working on omega-3 fatty acids, and India is also a major player, and Paris India Limited is one of the producer of omega-3 fatty acids from algae. And these are different products. Especially, I want to highlight these omega-3 fatty acids, beta carotene, astaxanthin. These are already available, and these are all derived from microalgae. And this Bonvita, the little champs, consists of DHA oil, which is derived from microalgae. This is a very popular drink or supplementary milk product which you use every day. So Little Champs has DHA, which is derived from microalgae. So already the application of algae in terms of industry is there, but still we have to understand the biology in order to understand what is happening. So here there is one more microalgae called Bioinformatics. This is basically a heterotrophic microalgae which can accumulate omega-3 fatty acids very fast, and it needs carbon source like glucose, galactose, and all. So in nutshell, we use different carbon sources and seen that glycerol may be the alternative source because glucose cannot be obtained, and glucose is required for fermentation. We need some alternative sources like glycerol or any other sources which can be used as a carbon, and so this can use glycerol as a carbon source. And you can see when it is in glycerol, you can see this light green fluorescent is the lipid body in glucose, and glycerol huge lipid is produced, whereas in other even lipid is produced, but growth is hampered. So it is very important to have lipid, and also growth should not be hampered. So we have done a reverse metabolomics approach here, and we have found the number of metabolites which are upregulated and downregulated. This is a nutshell of what happens with the metabolic profile when it is subject to glucose and glycerol, and we can see the arrows indicate different kinds of metabolites which are upregulating and downregulating. We also done some proteomics, and we laid with the proteomics data also dictates the blue and the red, which are upregulated and downregulated, and this also in very coherence with what we talk on metabolomics. Up to metabolomics is already published. This is unpublished data, so I'll be just highlighting what we are trying to see is this is the overall metabolic pathway regulation we see within the cell, where cell takes up glucose or glycerol, goes through glycolysis pathway, and from the pyruvate it diverts towards melanin, and from there are two different pathways, which is the fatty acid synthase pathway or PKS pathway, where they can accumulate omega-3 fatty acids. So there is a huge, so we are hypothesizing that there should be a alternative pathway, because omega-3 fatty acids is producing almost very in a high content in these cells. And why it is this is not only classical pathway, but also through PKS pathway. So in nutshell, this is how we work upon. And the 4th generation biofuels, this slide dictates how the energy sector is very important, and 72%, 72 to 73 to 75% is the one is huge energy. That's where in terms of transport or energy in industry or energy in buildings, all these comes under. So energy is a major area which has to be worked upon, and we need the energy as well. So this is a hypothetical 4th generation which can be aimed for, where a suitable microalgae can be engineered, or natural strain can be used to sequester CO2 from the atmosphere, and in the presence of sunlight, we can grow as a solar converter systems in an open system or industrial unit, which can be separated through, as I said, like there should be technology to separate your biomass to biofuels or other products, and the chemicals or fuels can be targeted. This is an overlay which is being proposed by Dual Technology, and this is the future of industrial biofuels. That's why it comes when you can sequester CO2, you are cleaning up the environment, you're cleaning the water through algae by converting the solar panels, and then converting that biomass into biofuels and byproducts. So finally, I would like to thank ICGB for supporting us in research, and also the external funding agencies like the different funding agencies helping us to do research, and my team, my Omics of Algae Group team, which we call it as GOA, especially without these guys like postdocs and PhD and project students, the work which I presented cannot be achieved. So with this, I would finally thank again my organizers, Professor Kumar, for inviting me for the lecture. And if you have any questions, I'm happy to answer.
Thank you, sir. It was an interesting presentation. So here are a few questions from the participants. I kindly request you to answer those questions.
Sure.
Which media are using to culture these algae? Is there any specific media or standardized to grow different algae?
Yeah, basically for omics experiment, we use specific media like minimal media. For example, what I am showing is marine media, which we use for microchloropsis or peak estuary. But at industrial scale, we are trying to scale up with the normal seawater. So you can take seawater and estimate what is the requirement of nitrogen and phosphorus in that, and you can substitute and you can use. But for omics experiment, we use minimal media and other enriched media to understand that. Yeah. Okay.
Okay, so thank you.
Yeah.
Another question is, which technology is using to decipher biofuel production pathway? What is the precursor enzyme or protein to genetically engineered pathway?
Okay, for biofuel production, there are many key proteins. So for example, DGAT, people have worked on diacyl glycerol transferase and glycerol 3-phosphate dehydrogenase and ACC enzyme, which is very important. And these are the key rate limiting. So you have to understand the physiology, how it is trying to accumulate. If it is upregulating, you can knock out and say that is a key regulating enzyme. You can overexpress those things and see that. But basically, there are different targets depending on what kind of biofuel is byproducts we are interested. For example, biofuels, people have worked on DGAT, and if you want to increase your this thing, you have to because there are some lipases which are very active, which can degrade the triacyl glycerol content. So you can knock down those lipases in order to increase the tag content. Also, is one of the approach where we can look for. Yeah.
Okay, so thank you. Another question is, any elicitors are using to enhance these biofuel production?
Yes. Yes. I didn't present one of our latest research what we published in Chemical Engineering Journal. So that is what we use different modulators or elucidators to enhance biofuels and biomass. So that is it's in a very like lab type of experiments are going on, but there are some modulators or enhancers which you can use to enhance the biofuel production. Definitely, that is what really the advanced research. We are also working on that thematic. Yes.
Thank you, sir. And last question, sir, is it possible to purify the air by this algae photosynthesis by installing in heavy air contaminated area? And this algae can used as raw materials in biofuel production. Is it possible, sir?
Yes, definitely. And this is what I am talking about, fourth generation. So you should have, instead of establishing a huge plant in order to sequester CO2, you should have a portable system arranged on the top of your apartment, just like you have Delhi, which is highly polluted. If you can place an algal system which can definitely decontaminate it, but these are the areas of future generation what I was discussing about, and people are working from different throughout the world to have this kind of what is called decentralized algal apparatus which can do the photosynthesis, and then once you get the biomass, that biomass can be used for biofuel. That's what algal systems not only produce biofuels, that they are always useful for production of other things also. Not only carbon sequestration, wastewater also can be used as a source of nutrition. So different kind of treatments can be done. Yes.
Thank you, sir. Thank you very much. Now I request.
Hello.
Yeah.
Hello.
Yes.
Very nice presentation, Pavan sir. I have one question.
Mm-hmm.
What is the cost effective of this biodiesel production, and what is sustainability in terms of, see example, biodiesel from JUTUR in Karnataka, already it is very somewhat, it is what you optimized, but it is not so up to the mark.
Yes.
So in that context, what is your opinion about of the sustainability and cost effective of and effectiveness also?
Yes. So definitely based on the raw material, the extraction process cost increases, especially now for algal biofuels. If you see the cost of algal biofuels, it is around $250 to $300 per barrel. But the crude oil is around $100 per barrel or 150. So we have to bring the cost of algal biofuels to make it sustainable. That is what it is. And as I said, like at this point, definitely it is feasible and also sustainable in due course of time, because many groups are working in that area. One important relevance, as I said, like people are working on high value products because you need some capex cost to be input in terms of biodiesel production. Because if you can add this high value co-products which are being produced along with biofuels or lipids, that will bring down the cost. And also at engineering aspects, we have to bring down the capex operational costs, especially when you have to separate algae from that. And with the JUTUR, it is especially difficult. Even it has a high lipid content, the extraction procedure is the one which is getting feasible. So they are, I think, first example of JUTUR oil is example of Indigo or Go Air flight, which has been flight by CSR Institute at IIP Dehradun. That is from JUTUR oil. So they are sustainable and feasible. But the thing is, people are now just trying to work out on high value products, so that once you have a cost from high value products, that can decrease the overall cost of your biofuel.
Okay, thank you, sir. Thank you.
Thank you. Thank you.
Thank you, sir. Now I request Mr. Rajshekar sir to thank the speaker.
On behalf of Department of Biochemistry, Davangere University, I would like to extend my sincere gratitude for your valuable time and informative lecture, sir. Thank you, sir.
Thank you very much. Thank you for giving this opportunity. I'm happy. Yeah, thank you.
Now I request Professor Wad. Now I request Professor Wadlapudi-Kumar sir to introduce our next session resource person, Dr. SHIVAKUMAR KEERTHIKUMAR, Peter MacCallum Cancer Center, Melbourne, Australia, on the topic Current Trends in Bioinformatics and Introduction to Sequencing Techniques.
Good morning, everyone. I'm very glad to introduce Dr. SHIVAKUMAR KEERTHIKUMAR. At Peter MacCallum Cancer Center, Melbourne, Australia. SHIVAKUMAR obtained his MSc degree from Department of Biochemistry, then Kuvempu University PG Center, and present Downgrade University. Dr. SHIVAKUMAR obtained his PhD from Institute of Bioinformatics, which had affiliation to Kuvempu University, Bangalore, India, under the guidance of Professor Akhilesh Pandey, Johns Hopkins University, USA. During his tenure as a PhD student, Dr. SHIVAKUMAR spent 1 year at Riken, Japan, as a part of Indo-Japan collaboration to work on his research project and gain substantial bioinformatics skills. Dr. SHIVAKUMAR then worked as a postdoctoral research fellow for 3.5 years at Radboud University, Nijmegen Center for Molecular Life Sciences, the Netherlands. During his tenure, he mainly focused on elucidating molecular mechanism involved in intellectual disability genes for better diagnosis of the disorders using systems biology approaches. During his career, Dr. SHIVAKUMAR gained extensive research experience in the field of bioinformatics, and later in 2014, he moved to Department of Biochemistry, La Trobe University, Melbourne, as a full-time research officer, and further gained research experience in understanding molecular mechanisms involved in secretion of exosomes using integrated genomic and proteomic approach. Currently, Dr. SHIVAKUMAR is a senior bioinformatician, where he is using high throughput genomics, transcriptomics, and proteomics research data to characterize cancer patients to identify molecular therapeutic targets. Dr. SHIVAKUMAR, using his bioinformatics skills, has significantly contributed his research work in the form of research papers published in peer-reviewed international journals, including Science, Nature Biotechnology, Nature Communications, the American Journal of Human Genetics, Class Genetics, Genome Biology, Nucleic Acids Research, Cell Reports, European Virology, eLife. He has presented his research work in many international and national conferences and supervised PhD and master research students. He has contributed 7 book chapters, 1 book, and 6 refereed conference papers. And with this brief introduction, I take this opportunity to welcome Dr. SHIVAKUMAR for this one-day webinar on Frontiers Biochemical Sciences. On behalf of Department of Biochemistry, Downgrade University, I welcome Dr. SHIVAKUMAR for this conference to deliver a lecture on current trends in bioinformatics and introduction to sequence techniques. Over to Dr. SHIVAKUMAR.
Thank you, Kumar sir, for the kind introduction. Can you all hear me?
Yes, yes.
Yeah, yes.
So let me start my talk by sharing the slide. So can you see my slide now?
Yeah, we can see you.
So is it in the full slideshow mode or?
Oh, I have to zoom it.
Okay, so then, now it's fine?
Yeah, perfect.
Okay.
You can continue.
Okay, so thank you, Kumar sir, for the kind introduction. And first of all, I would like to thank the organizers for giving me an opportunity to talk in this webinar, and also thank the organizer for organizing this webinar. So basically, today's talk, I'm going to focus mainly on the current trends in bioinformatics with emphasis to sequencing techniques. So I should get my slides at a very basic level because my target audience is mainly students, so that as Kumar sir said, maybe this talk will motivate young minds and PG students. Yeah. Yes, so I'm not going to spend much time here, as Kumar sir already said that I'm an alumni from 2001 batch. So currently I'm working as a bioinformatician at Biochemical Cancer Center. So this is the building where I work. Basically, the first floor of this building is mainly dedicated to cancer patients because this hospital also treats a lot of cancer patients, and the remaining floor is dedicated for research here. So throughout my career, I have gained an experience working on these techniques. So today I'm going to give a brief overview about these techniques, both at the level of genomics, transcriptomics, and proteomics. So I have so today's outline of my talk starts with bioinformatics. So I'll give you a brief introduction about what exactly is bioinformatics. So this would be useful for people who have never heard of this subject, and if they're interested, they can jump into this field for more details. Yeah, and then I'm going to talk on Human Genome Project, and then I'm going to talk on DNA sequencing techniques. So I'll give an introduction to DNA sequencing techniques and its application in the current field. And then I'm going to end my talk with giving an introduction to single cell sequencing techniques. And then if there's a time permits, I will also talk on my current research work. So let's see what exactly is bioinformatics. So the word bioinformatics mainly can be split into bio and informatics. So basically, informatics means study of computer science or information technology. And we know like bio is basically a biological science. It could be chemical science, it could be physical science, or it could be anything. So it's like a marriage between biology and computer science here. So that's what says bioinformatics. So in a simple term, it can be said like it's an application of computer programs in the field of biology. So it can also be defined as it's a subdiscipline of biology and computer science concerned with acquisition, storage, analysis, and dissemination of biological data. So in this context, my main focus will be on the DNA data, RNA, as well as protein sequences. So with this introduction to bioinformatics, I will just move on to now what exactly is this Human Genome Project. So the Human Genome Project was an international and collaborative research program whose goal was to complete and map the understanding of all the genes in the human beings. So this Human Genome Project was a 13-year-long publicly funded project. It was initiated in 1990 with the objective of determining the DNA sequence of the entire euchromatin human genome within 15 years. So when I say euchromatin regions, so these are the regions which you see more active genes transcribing, translating here. So, so just 2 years early, then their goal, they published a first draft of human genome in Nature journal in 2001. with sequence of entire genome 3 billion bases, so with some 90% complete. So mainly more than 2,800 researchers took part in this consortium and shared their authorship. So if you're more interested, you can go ahead to this paper and read more about the project. So the other interesting fact is the Human Genome Project generated enormous amount of data, which led to the growth of computational biologists and bioinformatics experts who brought the genome data to the widest audiences. So another important fact here is like it not only revolutionized the field of bioinformatics, but also gave me a new opportunity to try after completing my master's in biochemistry in 2001. So I still remember like I was in the final year of master's, and this particular draft was published, and there was a lot of news in the media as well as in the paper. And then I remember like I used to bug Professor Kumar sir, asking a lot of questions about what is this bioinformatics and all those things. So I think he said, I can say he guided me, and he said like, if you're more interested, why don't you go to a seminar and see what exactly is there. So he sent me to a seminar which was in Indian Institute of Science. And so I went there and I saw and heard this seminar for 3 days, I think so. And then I was more interested, and I decided that I'm going to continue my focus on this field. On this field, and then that is how I jumped into this bioinformatics field. So this is like, and also other thing is like, this is the first draft of the human genome was published in 2001. But this was not a complete sequence, because no one chromosome has been finished end to end, and hundreds of still unresolved gaps persist. And there are a lot of missing DNA sequences that are unknown. And most of these gaps were often contain repetitive DNA segments that were exceptionally difficult to sequence. And yet these repetitive segments include genes and other functional elements that may be relevant to human health and diseases. So during that time in 90s, there was no much technique available except Sanger sequencing technique, and which was very slow and time-consuming process. But slowly, I can also tell you the other important facts here. So another startling finding of this first draft was the number of human genes appeared to be significantly fewer than previous estimates, which ranged from 50,000 genes to as many as 140,000 genes here. Yes, and then, so when I say that draft and finished versions, the main difference mainly defined by coverage and the number of gaps and the error rate. So the draft sequence covered mainly the 90% of the genome at an error rate of 1 in 1,000 base pairs, but there were more than 150,000 gaps that existed in the draft version, and only 28% of the genome had reached the finished standard. But again, in 2003, they published another version which was having less gaps, and 99% of the genome is finished with an accuracy rate of less than 1 error every 10,000 base pairs. So if you look in the previous version, it is 1 in 1,000, and now it's like 1 in 10,000. So the error rate has drastically reduced here in 2003, because there was also improvement in the technology that was used to sequence this genome, which I will talk in my next slides. And again, last year, there was a consortium called Telomere to Telomere Consortium, and then they assembled a complete human X chromosome, and it was published in Nature, and it's known to be a landmark achievement for genomic researchers. Yes, so again, this year, last month, an international consortium has published a paper in bioRxiv. So this is still in the preprint stage, so it's not yet reviewed yet. They claim that they have finally sequenced the complete human genome, and they have added new 200 million newly sequenced base pairs. and 115 predicted coding genes. So I'm more interested now to see what exactly these genes are and do they play any role in disease-causing variants. Yeah. And then, so another important finding of this Human Genome Project research is that human nucleotide sequence is nearly identical. It's nearly 99% between any two individuals. And however, a single nucleotide change in a single gene can be responsible for causing human disease. So because of this, our knowledge of human genome sequence has also contributed immensely to our understanding of molecular mechanism underlying a multitude of human diseases. So currently, the reference genome that we have for human is also known as HG38 and HG19, and is used by essentially all researchers and scientists who need to align and assemble experimental or patient genome sequencing data. So it also serves as a consensus coordinate system for reporting results. So for example, if you have to sequence your own genome and if you get a sequence rates, you need to probably will end up aligning against this reference genome and see what is changed, what is normal and abnormal, and all this downstream analysis you can do by using this reference genome. So another interesting fact about this Human Genome Project is, so the reference sequence is not that one person, but it is a composite derived from several individuals. So therefore it is so this reference genome, what we call, it can also be called as a representative or generic sequence. So in order to ensure the anonymity of the DNA donors, what the scientists did was they collected more blood samples and were collected from volunteers than were used, and no names were attached to those samples that were analyzed. So thus, not even donors knew whether their samples were actually used or not. So yeah, and then basically this Human Genome Project was mainly designed to generate a resource that could be used for a broad range of biomedical studies. One such use is to look for the genetic variations that increase risk of specific diseases such as cancer, or to look for a type of genetic mutations frequently seen in cancerous cells. So more research can then be done to fully understand how the genome functions and discover the genetic basis for health and diseases. So give an introduction to Human Genome Project. And in the next slides, I'm going to talk about DNA sequencing techniques, which is mainly used to decode this Human Genome Project. Yeah. As previous talk, Pavan said that each sequencing technique can be spoken for more than an hour, but here I'm going to summarize this DNA sequencing techniques in try to simplify those techniques here. So first, let us see what exactly is DNA sequencing. So sequencing means determining the exact order of the base pairs in segment of DNA. So in which order this ATGC occurs, that's we call it as sequencing. So it can also be defined as the process of determining the sequence of nucleotides in a section of DNA. So the first commercialized method of DNA sequencing was Sanger sequencing. Probably you might have read in your biochemistry or in your master subject. It's also known as dideoxy or capillary electrophoresis based sequencing method. So this is a very old and original sequencing techniques, and it was named after who developed it. It's Frederick Sanger. Yeah. So it was a breakthrough that helped scientists determine the human genetic code, but it is also very, as I said in previous slide, very time consuming and a very expensive technique. But this Sanger sequencing method has been automated to some extent to make it faster, and it is still used today to sequence very short piece of DNAs, but it would take several years to sequence all of a person's DNA, known as individual person's genome. And then, so now, since this is a bit expensive and also time consuming, this is superseded by another generation of sequencing techniques. We call it as next generation sequencing technique, also known as high throughput sequencing. So it's all the catch term used to describe a number of different modern sequencing technologies here. You can say this is the first generation of sequencing technique, whereas this is second or next generation sequencing techniques. So the main difference here is like the Sanger sequencing can sequence a short fragment one at a time, whereas next generation sequencing technique can sequence millions of fragments simultaneously. So it's also known as massively parallel sequencing technique. So these technologies, as I said, it allows for sequencing of DNA and RNA much more quickly and cheaply than previously used Sanger sequencing, and as such revolutionized the study of genomics and molecular biology. Yeah, so using NGS, an entire human genome can be sequenced within a single day, whereas in contrast to previous Sanger sequencing technologies, that was used to decipher the human genome required over 13 years or more than 10 years to deliver the final draft. So that's you can see that there is a rapid evolution in the sequencing techniques now. So the next generation techniques, NGS thing also makes large scale whole genome sequencing accessible and practical for the average researchers because the cost is reduced, which I'm going to talk in my next slide. So it also enables scientists to analyze the entire human genome in a single sequencing experiment or sequence thousands to tens of thousands of genomes in 1 year. So if you look at the cost of sequencing this genome, as I said, the Human Genome Project took over 10 years and it costed them nearly $3 billion. So this graph here depicts the cost per human genome in the right side of this graph, and then the left side of my graph mainly depicts the cost associated with sequencing per megabase of DNA sequence. Yeah, so basically these two graphs depicts or illustrate the nature of reduction in DNA sequencing cost. So each graph here, it shows hypothetical data which reflects Moore's law. So that means a long-term trend in the computer hardware industry that involves the doubling of computer power for every 2 years. So in both the graphs, you can see like in y-axis is mainly the cost is depicted here, and in x-axis you can see the year-wise. So, but if you look in, again, the most common thing between these two graphs, it starts with 2001. So this is the year which the first draft of Human Genome Project was published. And then until 2008, you can see that the sequencing technique is still looks very pricey because they still were using Sanger sequencing techniques. Yeah, and then, but again, if you say starting from 2008, if you look here, in both the graphs, the cost drastically reduced because that is the cost of generating DNA sequence using second generation or next generation sequencing platforms. And the change in the instrument represents the rapid evolution of DNA sequencing technology that has occurred in recent years. So now, at present in 2020, you can also in the last year or in the current year, we can sequence a genome, individual's genome for less than $1,000. Yeah. So, so if you look at what exactly this, what are the techniques that are involved in next generation sequencing techniques? So I think in the previous talk, the speaker introduced to you about some of the Illumina sequencing ion torrent. And so I'm just going to give a brief overview about what exactly these sequencing techniques and their difference here. So basically there are different sequencing techniques, but these are most commonly used here. One is Illumina Solexo sequencing technique, and another one is the Roche 454 sequencing technique and ion torrent sequencing techniques. So basically all these NGS techniques relies on the basic concept that was used in Sanger sequencing, but slightly differs in the way it detects the and reads the sequence. Say, for example, if you look at this figure, it gives you an overview of DNA sequencing techniques. Say, for example, this is your gene of this is your tissue of interest. You extract DNA from this liver, you fragment them, you chop into different small fragments, and end of the rates will be there is something called adapters. They add to the end of the DNA fragments. And then they try to add to this kind of chip here or slides, which already has many oligonucleotide probes ready to get hybridized with the DNA of interest. And it's like each base, when it adds to the strand, it emits a fluorescent signal, and that fluorescent signal is read by the machine, and it reads the sequence. So in a similar way, so you have each of these wells here with all the reaction enzymes, buffers, everything like DNA polymerase. So it will simultaneously replicate thousands of fragments in a massively parallel way. And then the library prep is prepared and then fed into the sequencing machine, which can be Illumina, Roche, or Torrent. Yeah. So, and then once you get the data out, then again the bioinformatician comes here into the picture, and he will start analyzing all this data and other downstream analysis. So the basic difference here is like, so Roche 4-5 sequencing technique can be used if you're interested in sequencing very long reads, like say up to 1 kb base pair. If you're interested, you can go for Roche 4-5 sequencing, which is not possible in Illumina Solaxo sequencing techniques. And this technique is also known as pyro sequencing technique because it detects pyrophosphate release, again using fluorescence after nucleotides are incorporated by polymerase to a new strand of DNA here. So in the similar way, if you look at ion torrent or proton sequencing technique, it makes use of concept of release of H plus protons from the incorporation of individual bases by DNA polymerase. And then therefore it differs from the previous two methods as it does not measure the light. So both these techniques measures the fluorescent light, but this measures based on the H plus ions that are released. Yeah, so so then if you look at the applications of this next generation sequencing techniques, recently, I think the last month, the Technology Academy Finland awarded 2020 Millennium Technology Prize to two chemists, Shankar Balasubramanian and David Kleinmer. They are chemists from Cambridge University for the development of Solaxo Illumina next generation sequencing technologies. So this award was delayed by an year because of COVID situation. Yeah, and if you look at this application of this next generation sequencing technique, so this NGS technology has revolutionized global biological and biomedical research and has enabled the development of a broad range of related technologies, applications, and innovations, including helping in fight against COVID-19. So the next generation sequencing provides an effective way to study and identify new coronavirus strains and other pathogens. So currently this technique is also used here in tracking down which variant, whether it's an alpha, beta, gamma, or kappa, or delta variant. So with the emergence of the COVID-19 pandemic, the technology is now being used to track and explore mutations or variants in the coronavirus. So this work has helped the creation of multiple vaccines now being administered worldwide, and is critical to the creation of new vaccines against new dangerous viral strains. So the result will also be used to prevent future pandemics. So the technology is also allowing scientists and researchers to identify the underlying factors in individuals that contribute to their immune response to COVID-19. So this information is essential to unraveling the reasons behind why some people respond much worse to the virus than others. So in this slide, what I've done is because of time limitation, I have tried to classify these techniques at the level of DNA, RNA, and protein. So the figure that you see here mainly depicts the classic central dogma of molecular biology. Like you can see, this is a DNA fragment, and this is getting transcribed into an RNA and getting processed and exported to cytoplasm, where ribosome sits and then translate them into a protein here. So in terms of sequencing technique, at this level of DNA, you can see there are techniques which are known as whole genome sequencing techniques, exome sequencing technique, and chip sequencing technique. But at the level of RNA, there is a sequencing or transcriptomic technique, we call it, also known as bulk RNA sequencing or single cell sequencing technique. So I'll talk more on this technique in my next slide. And at the protein level, we have a technique called mass spectrometry. I think you also heard a bit of this term in the previous speaker. Yeah. So yeah, due to time concern, I'm not going to talk anything about this, but slightly I can give you some idea. Yeah. So let's give an example of DNA at the DNA level, how these techniques are used. Say, for example, let's take this exome sequencing technique here. So, and if you see at this image, you can see this double-stranded DNA genomic template as more of exons here. So these are the basically coding regions, and they are known to make nearly 1% of individual genome. So together, all these exons in a genome are known as exome. And the method of sequencing these regions, we call it as exome sequencing technique. And why this technique and how this is useful? Because you know that this is the coding regions, and this codes for protein or RNA, and any variation that you detect using this whole exome sequencing technique will help in identifying any disease-causing variants which lead to the cancer progression or any other diseases. So the whole exome sequencing technique is thought to be a very efficient method to identify possible disease-causing mutations. So let's come to this chip sequencing. So again, this is a combination of next generation sequencing technique with chip assay. Say, for example, if you look at this image here, it's a promoter region. So say if you're interested mainly in identifying genome-wide DNA binding sites, then you can use this chip technique. So basically, this is known as chromatin immunoprecipitation assay. So it's a powerful technique. So what exactly they do here is like the DNA bound protein is immunoprecipitated using a specific antibody. And then the bound DNA is then co-precipitated, purified, and sequenced here. So basically, so the chip-seq mainly helps in identifying genome-wide DNA binding sites for transcription factors and other proteins. So yeah, so it also helps us in gaining insights into gene regulation events that play a role in various diseases and biological pathways such as developmental and cancer progression here. But irrespective of any of the features, if you want to sequence the whole genome, then you can go for whole genome sequencing techniques, which is also very expensive, and yeah, it's a time-consuming process again. So yeah, so at the level of it's a level of RNA sequencing. You have a bulk RNA sequencing and single cell sequencing. When you say bulk, it's mainly if you isolate RNA from the whole tissue. But if you isolate an RNA from a single cell separated from that tissue, we call it a single cell sequencing techniques. As I said, like, I'll talk more on this in the next slide. Yeah. So if you're coming to the mass spectrometry based technique, which is at the level of protein. So basically, this is an analytical technique that measures mass to charge ratio of charged particles. And it's mainly used to determine the mass of particles for determine the elemental composition of any sample or a molecule here. So this has become mass spectrometry based proteomic analysis or study has become an indispensable tool for molecular and cellular biology and for the emerging field of systems biology. So widely used in quantification of proteins and peptides, protein-protein interaction studies, and post-translation modifications. Yeah. So let me move on to single-cell sequencing technique. So the big question is, why single cell? Why not you go for bulk RNA sequencing? What is the need for single cell? Again, it depends on the biological your biological interest and the question that you ask here. So, for example, this picture mainly depicts why single cell, why not bulk RNA sequencing technique. Say, for example, if let's assume that like you have a tissue of your interest, but you do the bulk analysis like without separating cell types, but you always end up doing getting average gene expression when you do the bulk RNA sequencing technique. Whereas if you do single cell sequencing technique, you isolate RNA from these single cell from different cell types, and then it will help in identifying cell subpopulation, which helps in understanding the heterogeneity. Say if you're working in tumor, we know tumors are mainly heterogenic in nature, and single cell techniques will help us in unrevealing those heterogeneity in tumor samples. Whereas bulk samples always marks the heterogeneity between the individual cells. And single cell techniques, as I said, it helps in understanding the heterogeneity, but also used to unveil cell subpopulation and the different gene expression. It can also be used to discover new cell markers and regulatory pathways. So uncover novel cell types, cell states, and rare cell types. So it also can be used to reconstruct developmental hierarchy, and you can do all kind of reveal, all kind of pseudo-time analysis using the single cell technique, which is not possible through bulk RNA sequencing technique. So basically, which technique you want to use, you need to ask yourself like what biological question that you want to answer from your sample of interest. Yeah, and then how we have different techniques in DNA, we have similar in single cell RNA transcriptome. We have different technologies, and I have classified them here based on broadly into three different categories. One is plate-based, droplet-based, and microwell-based. So in the platelet-based, there is something called SmartSeq and CellSeq. But I'm going to focus some more on 10x platform, because this is the platform we usually use in single cell technique in my lab. And I'll give a brief overview about 10x in the next slide. Yeah. So yeah, so if you look at this 10x Chromium or workflow of 10x single cell RNA sequencing, so basically this 10x uses a chip-like device. You can also call it as microfluidic chip. So this is the section of that microfluidic chip. So the main idea here is it has a lot of channels. So one of the channels that you can see here, I have zoomed in here. So what happens here is you have a lot of inlets here, like say your cells that have been separated into single cell, along with the reverse transcription reagents you can add here. And in another inlet, you can add 10x barcoded gel beads. So what exactly these gel beads, like if you see this figure here, you can see like this is a one gel bead, and it has thousands of oligonucleotide probes. You can see this particular probe here. And this probe basically contains poly-DT nucleotide here at this end, and which will actually hybridize to a poly tail of an mRNA from this cell. So the basic idea here is this single cell and this 10x barcoded gel beads along with the oil is captured in a vesicle-like reaction. They call it as GEMS, also known as gel beads in emulsion. So each functional gem that you can see the picture here, so this is one particular gem, where inside this gem, you can see one cell has been trapped here, And you also see the gel beads here, and also reverse transcriptase reagents here. And then there is a partitioning oil between different germs. So there is one germ here which will have another cell trapped inside the gel beads. Again, you will have another germ in which another cell will be trapped. So each single cell will be trapped inside this germ. So the next, what happens is inside this germ, a single cell is lysed, and it releases all its contents, and the gel bead is dissolved to free the identical barcodes, reverse transcripted oligonucleotides into the solution, and a reverse transcription happens with the polyadenylated mRNA thing. So then, once the cDNA, complementary cDNA is synthesized from a single cell, they will have the same barcode, like you can see this 10x barcode will be attached to those cDNA sequence. So this is like an address tag, like if you write in a letter and if you add a address to it. So that clearly shows that, okay, this particular cDNA is coming from this particular single cell. In the similar way, each of the cDNA is coming from each of the cells will have this address tag, so that we can go back, when we do bioinformatics analysis, we can go back and identify, okay, this particular transcript is from this particular cell, and this particular transcript is from that particular cell. So yeah, and then a library prep is done, and then it is fed into the machines for another bulk reaction. So in this slide, I have tried to summarize all the different techniques here. So as I said, it basically has a 4-step. Like any technique, like say it involves cell isolation, amplification method, sequencing method, and its application. Say for example, as I said, this drop-seq as well as scan-X, it mainly traps the cells inside the hydrogel beads. Whereas there are some techniques like cell-seq and mass-seq. So they use a machine called fluorescence activated cell sorting machine, which helps in sorting those cells, and then automatedly it will add it to the 96 well plates here, these techniques. And then there is also another technique which make use of this fluidic chip, which physically separates cells on this. So there are different way of cell isolation techniques here. But the other thing which you can also notice is the way it is sequenced. Like say some of these techniques like drop-seq, cell-seq, or 10x, they mainly sequence 3' end of the regions or 5' end of the regions. Whereas these techniques like SMART-seq, they sequence the full length transcript here. And again, like say, this is very, you have a lot of applications here, and again, it depends on the biological question of interest that you ask. Say, for example, if you're looking for 3' or 5' end sequencing, it's more accurate than use of unique UMIs here, but these techniques are much cheaper here. And depending upon your budget and funding, you can always go for this kind of technique. So in our lab, we usually use 3' end sequencing techniques here. And then do all the bioinformatics analysis to identify this gene expression thing. But in case of full length sequencing techniques, it mainly useful in detection of isoform level differences in expression. It can be used for identification of allele specific differences in expression. And so this technique will is also best for samples with a low number of cells here. Yeah. So given a slight introduction, now let's move on to these applications and tools involved in analyzing this single cell technique. Yeah, as I said in my previous slide, like some of these cell types are present in tissues which can be explored using single cell RNA sequencing techniques. And also it's used for elucidating the changes in gene expression during the differential process or across time or states. It can identify genes that are differentially expressed in particular cell types between conditions like treatment or disease conditions. So it can also explore changes in expression among the cell type while incorporating spatial, regulatory, and protein information. Yeah. So recently what happened is like this single cell RNA sequencing technique has been paired with genomics, epigenomics, spatial omics, proteomics, and imaging to achieve multimodal measurements of individual cellular phenotypes and genotypes. So in its purest form, we can say that the single cell multimodal omics mainly involves the simultaneous detection of multiple traits in the same cell. Yeah. And if you look in terms of tools that are used to analyze the single cell RNA sequencing technique, so some of the tools which are used for bulk RNA sequencing can also be used for analyzing the single cell sequencing technique. But in most of the cases, the computational analysis requires adaptation of the existing methods or development of new tools. So currently, if you focus on this image, you can see currently the number of tools available for sequencing is rapidly increased because the technique is also improving every year. And yeah, so you can see like currently we have around 1,000 tools that are available and developed for analyzing these single cell data. And in terms of publication, you can see among them, there are like more than 50% of the papers have been published, and still there are some in preprint and some not published. So in terms of tools, like you can use, as a bioinformatician, you can use many language and to develop these tools. Say, for example, if you look among these 1,000 tools, you can see majority of the tools were developed using R package. It's a free and also it's a statistical package. And then the next around 32% of those tools were also developed in Python scripting languages, and rest of them like C++, MATLAB, and other things. So basically in my lab, I basically use these two languages or these two packages to analyze and analyze the single cell technique. Yeah. So and I think it's a very good opportunity for the bioinformaticians or any PG student if are interested in bioinformatics to learn these new techniques and languages to analyze this data. Or you can also end up developing new tools and resources to aid the analysis, because this is the field which requires basic knowledge in biology, stats, maths, and other programming languages. Yeah. So I think, do we have some more time? I can talk about my current research work. Hello?
You can proceed.
Yeah, okay. So can I talk? Yeah. Okay, I will take it as yes, and I will, I'll give a slight introduction to my current research work. Okay, so let's assume, so let's talk about my research work here. So basically, the current lab mainly focus on prostate cancer project, and say this is a cancer patient with a tumor, and what we do is like we extract the tumor tissue from the patient, we collect, and then what we do is we graft them into the immunodeficient mice, and we generate a PDX models. And then what we do, and we allow this tumor to grow inside the mice for a certain period of time, and then we apply different treatment condition on these PDX models here. So what exactly we do is like we test different drugs, and we test the efficacy of these drugs, and we also see like what is the response of this drug, whether the tumor has decreased or not. And then sometimes instead of individual drugs, we also try different combination of drugs on these mice and see if there's any tumor volume decrease or any increase in the drug response. And then this is done, and then we extract again those tumors from the mice, and then we apply these techniques like whole genome multilevel genomics. We do targeted sequencing, exome sequencing, whole genome sequencing, and chip sequencing. And then we also do single cell and bulk RNA sequencing. And then also we do mass spectrometry proteomic-based analysis to answer different questions and characterize these clinical PDX models. So what basically, once this is all done in the wet lab, and then the data comes to me, and basically I do a lot of data analysis using these techniques, the data from these techniques, and then not only just do the data analysis, I also what do is like try to store this data, because our ultimate aim is to share this data to the public so that other researchers can use and do some downstream analysis. And then if there is a need, then we also end up developing any tools here. Yeah. So this is what briefly we do in our lab. But I also try to I have given some, like say in this slide, I have selected some of my research paper that I've published, and I've tried to categorize them based on the type of data or analysis that I do. Say, for example, as I said, just do data analysis from those techniques, or develop tools or software package development, and or anything related to data storage and database development. So if you look at these papers, so each paper here is unique, and we have used different techniques, like say for example, in these two papers, we have used systems biology approach and just did the data analysis, and then we have published this paper here. And in this paper, I have done RNA sequencing analysis technique here. And this one paper is basically, we have data from high throughput mass spectrometry based proteomics data. And this paper, which published last year in Science, we have done a lot of whole genome sequencing thing here. And then this is like last week, it recently got accepted in Nature Communication. So we have done basically here proteomics as well as RNA sequencing techniques. So if you're interested, please go to this paper and read more about to get an idea of what exactly bioinformaticians do and how real-time data can be used here. So there is also some paper where we have developed a tool, like there is something called FUN, which is a functional enrichment analysis tool. And there is something called another package which was developed in R for next generation sequencing technique. And this is a web-based visualization tool for analyzing mutation from human genetic disease. So this is basically a tools paper. Yeah. And when you come into database storage or web-based database development, we have developed almost many, but these are a few selected ones here. One is called Rapid and Human Proteinpedia and NetPath. Yeah, so if you're more interested, please go to this paper and see and get more details about it. Yeah. So I think I've come to the end of my talk. Thank you very much. And thanks for listening to my talk. And I hope you might have find these details useful. And if you have any questions, please do not hesitate to contact me. And I assume my today's talk would have given you all overview of the current trends in bioinformatics with emphasis to various sequencing techniques and challenges. So if there are any students studying masters in biochemistry or microbiology, or any PG students in today's audience and who are still thinking what next and what is the future and which area you want to focus, I would definitely encourage you guys to try this fast growing and challenging bioinformatics field. Yeah. So once again, I thank the organizers for organizing this webinar and giving me an opportunity to talk in this webinar. Thank you. Yeah.
Thank you, sir. It's very informative session. Here are a few questions, sir.
Yeah.
What are the social issues related to the Human Genome Project?
So do you mean ethical issues?
Yes, sir.
No, that the social ethical issues, there is nothing like actual social issue, but it's always a stigma associated with that. Say if you are like say you want, it's like you are trying to sequence your own genome. There's always an ethical issues. You can't make it more public. Some has an issue because I have an experience. When I was a PhD student, we had a collaboration with a private company. I don't want to name that company, and we sequenced genome of two celebrities. One is the IT guy, another one is a Bollywood guy. So, but but we couldn't publish because there were a lot of issues with it. There's still issues, but I think this can be taken care in the initial stages. Yeah.
Okay, so thank you. So another question, is sequencing help to develop a drug against a particular cancer type?
What is that? Is
sequencing help to develop a drug against a particular cancer type?
Yeah, because like I showed in my current research work slide, because we test the efficacy of the drug and see how the person's genome has responded to that drug, and which will help in identifying what variants are there, and we can try to customize the treatment therapies. Yeah, it can be used. Yeah.
Okay, thank you. And last question, sir. To what extent these techniques are being used in development of personalized medicine?
Yeah, it is being used. I think, I don't know, like some of you guys might have heard of Angelina Jolie thing happening, and she did a double mastectomy thing. So yeah, I think it's quite gaining rapidly. It's like people are now focusing on the person genome thing. But yeah, I said like there are a lot of still ethical issues with that, and also the costs associated with that also. Yeah. So you can. It's still evolving, and people are trying that personalized medicine thing. Yeah.
Okay, so thank you. Now I request Professor VENKATESHWAR sir to thank the speaker.
Yeah, let me stop.
Dr. SHIVAKUMAR, thank you very much on behalf of the Department of Biochemistry and Downstream University. Thank you for your time and sharing your experience and encouraging our students to plan their career in future. Thank you once again. In spite of your busy schedule, and we could make it to be amidst us online. Thank you.
Yeah, no worries. Thank you, sir. Yeah, it's always welcome. Yeah. Yeah, it's good. Yeah, it's good to talk and share knowledge and give back to the community from where you are here. Thanks.
Thank you. Thank you very much. Thank you.
Now I request Professor Gopal M. Advirao sir to introduce next session speaker, Dr. VENKATA DK, CSIR CMAP Research Center, Bangalore, on the topic role of lipids in signaling mechanisms.
Okay. So good afternoon to all of you. The Honorable Vice Chancellor, Professor S.V. Halse sir, Professor Anita H.S., Chairman and Professor of Biochemistry, Dr. VEDLAPUDI, and this session speaker, Dr. VENKATA DK, and all my colleagues, invitees, and all participants. It is my great the pleasure to welcome you all for this one-day webinar organized by the Department of Biochemistry of Davangere University, Davangere. So here I will take this opportunity to introduce the speaker of this session, Dr. VENKATA Rao. Dr. VENKATA Rao is a senior scientist at CSIR, Central Institute of Medicinal and Aromatic Plants, Research Center, Bengaluru. He obtained his MSc degree from Gulbarga University and PhD from Sri Krishnadevaraya University, Anantapur. Dr. VENKATA Rao was a senior research fellow, as well as he worked as a research associate at the Department of Biochemistry, Indian Institute of Science, Bangalore, during the period 2007 and 8. He was the postdoctoral fellow at the same Department of Biochemistry, IISc, Bangalore, during 2008 to July 2009. He also served as a scientist fellow at CSIR, CIMAP Research Center, Bangalore for 2 years. Dr. Venkata Rao, the current area of research interest is lipidomics of yeast and plants. He has a rich experience as a senior scientist with a demonstrated the history of working in the biotechnology industry. And he has skilled in synthetic biology as well as bioinformatics. Currently, Dr. Venkata Rao has been working on design, synthesis, and the validation of DNA cassettes for yeast engineering to produce high-value terpenoid compounds. Dr. Venkata Rao has many research projects funded by various funding agencies, and he has authored more than 50 research papers in national and international reputed journals. He is a faculty member of ACSIR, Academy of Scientific and Innovative Research, and he is a member of several professional societies both in India and abroad. With this brief introduction about the Venkata Rao, now may I request Dr. Venkata Rao to deliver his talk on role of lipids in signaling mechanism. Over to Dr. Venkateshwar.
Thank you, Dr. Adavi Rao, for your kind introduction about me and myself, Venkat Rao, currently working in a Central Institute of Medicine and Aromatic Plants. So can you hear me all?
Yes.
Yeah, I'll try to share my slides now.
Yeah, I can.
Can you able to see the full screen?
Yes.
Yeah. Thank you so much, Dr. Vadlapudi Kumar, for inviting me this webinar on frontiers in biochemical sciences, and supported by Davangere University and also the SBC and the Davangere chapter. So before going to the details of my talk, I'm very much fascinated with two earlier talks. First is on omics of algal research. It's a wonderful talk, particularly on how to increase the triacyl glycerols in algal system. Then followed by SHIVAKUMAR, his best friend of mine. We had a short time of colleagues in Bangalore. So where we have some kind of a training in bioinformatics fields. Then I moved to CSIR system, then he moved to abroad for his further postdoctoral studies. With a small introduction, I would like to give why the lipids are so important. You know, right, there are, if you look at the biological system, there are three important biomolecules. One is proteins, carbohydrates, and followed by interesting molecule that is called lipids. Lipids, apart from proteins and carbohydrates, this is highly hydrophobic in nature. Then these lipids play a very vital role in transmitting various signals from external stimuli, whether it's a plant or human or any kind of a microorganisms. With this small introduction, I'll just move on to the second slide. So these lipids, one second, yeah. These lipids have been broadly categorized into neutral lipids. It's mainly stored in the form of oils, and whereas in humans, it is in the fat. And the phospholipids, they are very important role in membrane biogenesis. Particularly, the different types of phospholipids are involved in dynamic nature of the membrane. So these membranes play a vital role in reacting to your body system. When there is a kind of a signal that is going to transmit immediately to the cells to react to withstand that kind of stress conditions. There are two pathways have been deciphered in biological system. One is the cytosolic triacylglycerol pathway. It is completely independent of phospholipids. Whereas the other one is a membrane bound. It is bound to the endoplasmic reticulum. If you look at the cytoplasmic triacylglycerol pathway, triacylglycerols are basically synthesized from glycerol 3-phosphate, which is then acylated to lysophosphatidic acid. These lysophosphatidic acids are again acylated in the in presence of lysophosphatidic acid phosphatase to produce monoacylglycerol by dephosphorylating phosphate from the lysophosphatidic acid, which is then acylated to form a diacylglycerol, and finally it is going to form a triacylglycerol, which is actually inert molecule. It's going to store in the form of fat or oil. This is also known as monoacylglycerol dependent pathway. On the other hand, there is a very important pathway called membrane bound pathway. This also synthesizes triacylglycerol from glycerol 3-phosphate to lysophosphatidic acid. Whereas in case of lysophosphate, there is an enzyme called LPA acyl transferase. It's going to produce more of phosphatidic acid. This phosphatidic acid takes another route called PA phosphatase that is going to synthesize diacylglycerol to produce triacylglycerol. Why I am stressing the phosphatidic acid and diacylglycerol? Phosphatidic acid is a central molecule for both membrane biogenesis and neutral lipid biogenesis. Whereas in case of cytosolic, it's mostly in the oil plants, we can see only, without phosphatidic acid, we can see only diacylglycerol and triacylglycerol synthesis. So apart from its role in triacylglycerol biosynthetic pathway, it is PA play a very vital role in biosynthesizing various phospholipid molecules. One is called CDP dependent pathway, where we can see the synthesis of phosphatidyl inositol. All these highlighted phospholipid species play a very vital role in membrane dynamics. When this CDP diacylglycerol is converted to phosphatidyl glycerol phosphate, which is then followed by producing phosphatidyl glycerol. On the other hand, this also the two molecules of CDP and phosphatidyl glycerol to form caudiolepins. On the other hand, this phosphatidic acid is converted into diacylglycerol in presence of phosphatase activity. This phosphatase activity, then it is going to convert into phosphatidylcholine, which is a very important major phospholipid species in membranes. This is further converted into phosphatidylethanolammine, and finally it is going to give serine. These are diverse array of different phospholipids. They play a very vital role in membrane dynamics. See, any changes, any alterations in the composition of these particular phospholipids, that will bring a lot of changes in our body, or in a plant, or a microbes, or in a human body. We can see a lot of kind of disorders, metabolic disorders. So this homeostasis has to be maintained based on the various signalings. If the the PA levels are very high in a system, that's going to result in some kind of a genetic alteration in the system. Where it is low, there is a kind of a diseases related to the lipids. So the phosphatidic acid play a very vital role in both biological and physiological system. PA is one of the simplest group of glycerophosphoric acid. It's a very simple, it has two acyl chains and one carboxylic group. And this phosphatidic acid is essential for phospholipids and neutral lipid biosynthesis. And it is a common precursor, very important, is the only common precursors that is going to decide where the cells have to synthesize, whether it's a membrane or going for a energy storage molecule. So in case of triacylglycerol biosynthesis, phosphatidic acid is dephosphorylated to produce diacylglycerol. In presence of phosphatidic acid phosphatase, this undergoes of dephosphorylations to produce a lot of diacylglycerol, which eventually converted into triacylglycerol. If cells want more membrane, it is going to convert, the PA is converted into CDP diacylglycerol to produce more of CDP DAG, that leads to the production of more of different phospholipids. This is catalyzed through cytidine transferase activity. PA is a key intermediate metabolites that decide which pathway it has to take. So cells have a kind of a mechanism that is going to decide whether the cell has to synthesize the energy or it has to go for membrane. If cells feel there is a lot of energy, the cells start expanding by storing more amount of lipids. So as a result, it stops synthesizing the storage lipids, then it goes for a membrane. Because cells start expanding, it requires more membrane to synthesize. So that is the reason there is a kind of a unique mechanism that operates to regulate the phosphoric acid levels in the body. However, what kind of factors that communicate to the phosphoric acid, whether it has to go to neutral lipids or phospholipids, it is still a debatable question. So there is a lot of scope in understanding in plants and microbes and human system to understand the disease and metabolic disorders. If you see the right side of your figure, if the glycerol 3-phosphate is converted to lysophosphatidyl, then further it is converted to either storage lipids or membrane lipids. This PA flux, the endogenous PA flux, the levels of PA is commonly regulated by an enzyme called cyclin-dependent kinase. This cyclin-dependent kinase, when there is an excess of triacylglycerol, it is going to phosphorylate simultaneously two enzymes called triacylglycerol lipase and also the PA phosphatase. The phosphorylated PA phosphatase becomes inactive, so its inability to synthesize diacylglycerols because it is inhibited. Since the cells feel there is a high amount of triacylglycerol, it undergoes triacylglycerol hydrolysis that results in more of diacylglycerols. It will go into various pathways like phosphoric acid pathways. So this is one of the important crucial CDK activities is very crucial to understand how this kind of a senses, whether how the CDK1 and DAG and PA, these three play a very crucial role in the activity of cyclin-dependent kinases. However, there is no exact mechanism is known for this particular activity. And PA signaling is very important for mTOR pathway activity. mTOR is an important activity, is crucial for cell division during growth and development. And PA is also involved in activating various other metabolic pathways like amino acid biosynthesis and uptake of various nutrients. Actually, mTOR is highly stable in presence of PA. When PA is bound to the mTOR, that complex is highly efficient in the activity. So PA sensing is essential for mTOR because during cell growth and development, mTOR has to be communicated that there are lipids which are present in the system, then cells start dividing into many multiple cells. In that case, this kind of signals, there are limited amounts of lipids are there, cells can go for the division. In that cases, PA play a very vital role. PA senses the mTOR that there is a sufficient amounts of lipids are present for the further membrane biogenesis during cell division conditions. These phosphoric acid play a very vital role in key signaling. This signaling mechanism is operated through PA by two distinct mechanisms. One is by the membrane-bound phospholipase D activity. Phospholipase D activity takes some amount of phosphatidylcholine to produce directly the phosphoric acid. This phosphoric acid transmits the signals from membrane outer environment to the nucleus. On the other hand, there is one more activity called PLC-DGK1 activity. In PLC-DGK1 activity, PLC plays a very vital role in producing more amount of diacylglycerols. The amount of diacylglycerol is converted into phosphoric acid by DGK1 activity. These two mechanisms are independently operated or regulated to generate more amount of phosphoric acid. Phosphoric acid PLDs and PLC-DG activities are involved in various signaling mechanisms. So PA plays a mediator role to transmit the signals. So without PA, there is no life. So PA must be there in a system. Without PA, there is no any kind of a transmissions. So this PA acts as a biotic and responses to various stress conditions like biotic and abiotic stress conditions. These two activities, PLDs and PLC activities, are very important during cell signaling mechanisms. Apart from its role in the synthesis of various phospholipids, PA can synthesize phosphatidylinositol cardiolipids. On the other hand, diacylglycerols are going to synthesize phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine, and finally triacylglycerol. The homeostasis of phosphatidic acid and diacylglycerol is mediated by two crucial enzymes called diacylglycerol kinase and another one is PA phosphatase. Any alteration in this particular homeostasis resulted in lot of abnormalities in the cells. Apart from its role in biosynthesis of various lipid compounds, it acts as a second messenger, like activating mTOR, and it also involved in amino acids, PA binds that I have already discussed, and it plays a very vital role in mTOR activity. PA levels are very high in cell systems when cells have various abiotic stress conditions, kind of heat, cold, or any soil stress conditions. When the plants or any system is attacked with like insects or a kind of infections, the PA levels are high. This clearly indicates that whenever there is stress, there is a high amount of PA is accumulated that is going to modulate whole your molecular system to respond to that particular stress signals. And it is PA levels are very high when you have artificial stress hormones like salicylic acid and abscisic acid. And during plant development, PA levels are very important. So the PA is regulated by two different mechanisms. When there is stress from the outside, there is receptors. That receptors, whether physically or physiologically, binds to the PLDs, phospholipase D. This phospholipase D, the activated form of phospholipase D goes to the membrane where it takes some amount of phospholipids to produce more amount of phosphoric acid. But how this phosphoric acid is translocated from the cytosol to nucleus, it's still a debatable question. The another signaling mechanism that is operated through diacylglycerol mediated signaling response. Similar to earlier slides, when there is a signal, there are some receptors that's going to activate phospholipase C. Similar to phospholipase D, phospholipase C is going to take some amount of phospholipids, phosphatidylcholine. Instead of producing phosphatidic acid, it produces diacylglycerol. But here is a question whether diacylglycerol is converted to phosphatidic acid or diacylglycerol itself is going to the nucleus and do and will bring some kind of a response to that particular stress. These are still debatable questions whether PA, phosphatidic acid or diacylglycerol are involved. In order to understand these signals, we have done some simple experiments. We have done some simple TLC methods to understand whether PA is involved or diacylglycerols are involved in ergosterol biosynthetic pathway. So why we need to study the lipids? As we know that lipids are very major molecules during cell signaling process. So understanding how these lipid modulations that's going to help or regulate metabolic pathways such as secondary metabolism. Since I am in this institute, we are particularly working on high production of triterpenes from yeast actually. So we are trying to modulate lipid pathway to understand whether is there any effect on the production of secondary metabolites. See, many people are trying to do complete pathway engineering to produce the huge amount of the compound of interest. Instead, we can do say small alteration in lipid metabolism can be able to produce more amount of target molecule of our interest. So we have recently identified a novel mechanism that's going to produce a huge amount of the compound of interest that belongs to secondary metabolites. So it is better to fine tune this kind of pathways rather than whole pathway engineering to do small modifications in lipid pathway to gain more amount of the desired molecules, to alter the whole system to fine tuning for the production of our compounds. So develop and design the yeast strains for the bioproduction of our natural compound. This is a major work is going on in our laboratory. So there are two important lipids that play a multiple role, both phosphatidic acid and diacylglycerols. So let us start with my work where we started to understand how these lipids are involved in regulating ergosterol biosynthetic pathway. The ergosterol biosynthetic pathway starts from the mevalonate pathway. Some of these intermediate metabolites are very important they are very important as important metabolite precursors for various high value compounds. For example, sesquiterpenes. Sesquiterpenes are produced from the farnesyl pyrophosphate. Farnesyl pyrophosphate is a part of ergosyl biosynthetic pathway. For example, squalene. Squalene is a second intermediate metabolite for the production of plant-based natural triterpene compounds. So why don't we understand, why don't you alter these metabolic pathways to have a more amount of ergosterols? So apart from the lipids role in storage, energy storage, and membrane biogenesis, lipids are known for various biological processes like signaling and proliferations. The diacylglycerol and phosphoric acids are well-known signaling pathways. However, their exact mechanism is still unclear. We use yeast as a model system to study the physiological role of these particular signal lipids with respect to the ergosterol biosynthesis. So most of these pathway genes are characterized in lipid pathway. To understand its role, signaling role in myelinate pathway, we have done set of gene deletions in yeast. As a first step, we have deleted 2 genes. They are called diacylglycerols. This is a wild type. If you look at here, this is a wild type. There the endogenous levels are normal. When you delete the 2 acyltransferase, we hypothesize that there is a huge amount of diacylglycerols are present. So we are endogenously increasing the signaling molecules by deleting the acyltransferase activities. And we have also done some other mutations where there is a reaction called phosphoric acid to diacylglycerol. So when you delete of PAP activity, PAH1 activity, that's going to give a more amount of endogenous phosphoric acid. So we took these 2 phenotypes to check is there any effect on the ergosterol metabolism. That is also called myelinate pathway. So our initial mutant screening demonstrated the involvement of different lipids in MEA pathway regulations. The yeast cells lacking diacylglycerols exhibit altered ergosterol levels because ergosterol pathway and lipid pathway both are independently operated. But how these phospholipids are regulating the ergosterol is still a debatable question here. Then initially we have done real-time data demonstrated that when we have a more amount of diacylglycerol or phosphoric acid, we see a elevated MEA pathway activity. Further, also we have quantified the various metabolites like squalene and ergosterol, and we see a more amount of squalene and ergosterol. These are the end products, important metabolites of ergosterol pathway. So diacylglycerol and PA homeostasis is mediated by diacylglycerol kinase. So when you delete diacylglycerol acyltransferase, then what is the fate of diacylglycerol kinase activity? So our preliminary data convinced that to some extent whether PA or DAG may be involved in myelinate pathway regulations. But however, we are not sure which metabolite is involved. So when we delete diacylglycerols, we see more amount of, we quantified, we see more amount of diacylglycerols. There we took these 2 cell phenotypes. When we analyzed the ergosterol levels, we could be able to see more amount of ergosterol as compared to the wild type. This clearly demonstrated that somewhere there is a link between these 2 pathways, phosphoric acid and the ergosterol, though they are independently operated. So altered DAG and PA homeostasis in diacylglycerol acyltransferase knockdown that leads to high accumulation of ergosterol as compared to the wild type. When you delete acyltransferase, we could be able to see more of ergosterols. So we went ahead and did some kind of a GC chromatogram. If you look at the left side of your screen, this is a third peak number 3 is ergosterol and peak number 1 is squalene. When you delete DGA, diacylglycerol, we could be able to see more accumulation of ergosterol as compared to the wild type. This clearly demonstrated that the lack of diacylglycerol, the lack of the cells lacking triacylglycerols, it is going to trigger the ergosterol pathway through DG or PA levels. So our GC and TLC analysis proved that the yeast cells are found to substantially accumulate squalene and ergosterol. These are the key metabolic indicators of melanate pathway activity. If you want to check the melanate pathway activity, you just need to quantify squalene and ergosterol. That is going to give a basic information how your cells are affecting when you delete a particular activity. So then we looked, when we delete diacylglycerol activity in the yeast cells, we could be able to see the high expressions of the important melanate pathway, ergosterol pathway genes. That is ERG10, HMG, ERG20, ERG9, and ERG1. And we also complemented these knockouts with its functional acyl transferases. We could be able to see there is a drastic reduction of ergosterol pathway genes. This clearly demonstrated that somewhere there is a signaling mechanism that is regulating melanate pathway, whether it's a diacylglycerol or phosphoric acid. So when we delete acyl transferases, then we ask two questions: whether DAG is more or phosphoric acid more, and we also looked at DAG kinase activity. So we initially assumed that when we delete diacylglycerol acyl transferases, we thought that there is a more amount of diacylglycerol. Instead, we found more of phosphoric acid, and then we looked at diacyl kinase activity, and we see the more expression of DGK1. It is possible that when the cells having more amount of diacylglycerols, it is converted back to phosphoric acid. This phosphoric acid may go for membrane biogenesis. So DGA1 mutants have ability to mobilize excess amount of diacylglycerol to phosphoric acid through DGK1 activity. However, how this DGK1 is upregulated is still unknown. Still needs to be studied further. So there is an excess amount of phosphoric acid is resulted in more amount of ergosterol biosynthesis. Then what we did was we just overexpressed the diacylglycerol under strong inducible promoter DGK1. When we overexpressed diacylglycerol kinase, we still see the more expression of ergosterol biosynthetic gene. This clearly demonstrates that diacylglycerol kinase activity is very crucial for upregulating the phosphoric acid. This more amount of phosphoric acid is very important for regulating the ergosterol or melanate pathway. Then we also quantified the phosphoric acid levels and as well as DAG. When you have more amount of DGK1, we could be able to see more amount of phosphoric acid. This is a phosphoric equivalent, this is a phosphoric acid. It's very difficult to capture. Then we finally got with many experiments, and we also looked for diacyl and as well as total phospholipids and PA levels, and we see a significant increase in the total phospholipids. And we also looked at diacylglycerol levels are very high. When you express DGK1, we could see there is a drastic reduction of diacylglycerol as compared to the wild type and DGA1 and LR1 knockouts. So when you overexpress diacylglycerol, then we look the fate of the squalene levels. As we know, there is a high expression of ergosterol biosynthetic pathway. We could be able to see the drastic increase in the squalene level. There is a peak number 1 indicates squalene, peak number 3 indicates ergosterol. So when you overexpress diacylglycerol kinase in DG deficient mutants, we could be able to achieve the more amount of squalene. So the squalene levels having compared is almost 6 to 7 folds higher as compared to the wild type. This clearly demonstrated that can we use this system to produce a more amount of triterpenes. So squalene is one of the important triterpene molecules widely used in cosmetics, medicines, particularly in the development of vaccines. So why don't you divert this squalene, huge amount of squalene that we produce by our metabolic engineering strategy, to have more amount of triterpene, that is called beta-amarin. So beta-amarin is one of the important compounds. So then we look when we overexpress a plant-related gene that is called beta-amarin synthase under in PGAL DGK1 cells. When you overexpress beta-amarin synthase, we could be able to see the huge amount of beta-amarin accumulation as compared to the wild type. peak number 4 indicates a beta-amarin. Beta-amarin is widely used in various medical applications. Like it acts as an anti-cancer and anti-inflammatory molecule. From plants, it produces in a very minute amount. In yeast system, we are able to produce a huge amount of beta-amarin from the plant. So we also overexpress the beta-amarin synthase in DJ, diacyl glyceroacyl transferase knockouts, which is overexpressing DGK1 one background. Still, we could be able to see the huge amount of beta-amarin production as compared to the wild type, and as well as beta-amarin alone. So this almost like beta-amarin production was almost like 30 micrograms per ml as compared to the wild type. It's around maybe 2 to 3 micrograms per ml. This clearly indicated that this is going to be very sustainable and economical approach to synthesize any kind of a triterpene molecules which are interested, which is very difficult to isolate from the plant sources. So this kind of modulations, metabolic modulation, that's really going to help to fine-tune your metabolic engineering, metabolic process to produce your compound of your desire. The another one, the knockout that we worked on is PAP. PA phosphatase knockout has been and that is going to, so when we knock out PA phosphatase, there the excess amount of the cells are inability to convert phosphoric acid to diacylglycerol. So here also we see a more amount of phosphoric acids, and here also we could be able to see more amount of ergostable formation as compared to the wild type. This is another kind of an evidence that when there is a high amount of phosphoric acid, we could be able to see more amount of ergostable. We also looked at mRNA pathway transcript levels. There we could be able to see the high expression of different ergostable pathway genes like ERG10, HMG, ERG20, ERG9, and ERG1. We also quantified ergostable levels and as well as squalene. There, it has been demonstrated that there is a huge accumulation of ergostable and end product of ergostable biosynthetic pathway, squalene as an intermediate of ergostable pathway. This clearly demonstrated that there is a high amount of PA, there is a high amount of squalene. This squalene can be diverted to your high value compounds. And we also did whether the DGK1 is involved or not. When we knock out the PA phosphatase, we see more. Then we complement with both functional PA phosphatase and also catalytically inactive. Whether PA phosphatase is involved or not, we transform the cells with a functional PA phosphatase and also a phosphatase which is catalytically inactive. Even if you transform, you don't see the PA phosphatase activity. When you transform it, when you transform the PA in inactive PA phosphatase, still we could be able to see more. It's similar to PA phosphatase. When you transform the functional phosphatase, PA phosphatase, we could be able to see a clear reduction of ergostable. This clearly indicates that PA phosphatase activity plays a very vital role in regulation of melanate pathway activity in the cells. And we also quantified and we observed there is a drastic increase when you quantify the squalene level. There is a drastic reduction when you transform the cells with PA phosphatase. Further, we have also quantified the melanate pathway transcript levels in both PA phosphatase knockouts and PA phosphatase knockout, which are transformed with a catalytically inactive PA phosphatase, that is PAH1D. It's a mutation at aspartic acid 2 alanine and aspartic acid 2 again alanine. There are two important catalytic residues are mutated to inactive amino acids. So these cells, we could be able to see there's a more amount of transcript levels are observed in PAH1 knockout and also PAH1 knockout transformed with catalytically inactive. Whereas in case of PAH1 transformed with the functionally active PAH1, there is a clear reduction of MVA pathway activity. This clearly demonstrated that PAH1 plays a very vital role in regulation. So, based on our reports, so we are the first one to show there is a crucial role of diacylglycerol kinase and PA phosphatase that play a very vital role in melanate pathway regulations. Overexpression of diacylglycerol led to high squalene and intercostal flux through PA signaling. So DGK1 overexpression that leads to high PA levels. If you have high PA levels, so what we did was we co-expressed DGK1 and beta-amarin synthase from amarin beta-amarin synthase. We could be able to see the high amount of beta-amarin production. So this is kind of a novel strategy that we proposed and published in BBA. This work done by Purnima. Now she is in US as a research scientist at Kurishima Bio, a Johnson & Johnson company. So finally, other than its role in PA biosynthesis, we are able to, and many researchers have done wonderful work in plant signaling of PA, how this PA is involved. And these PAs are also involved in giving strength and cell elongation and also the mechanical strength to the plant. Lee et al. In 2011, they have discovered a gene called PLA3, which physiologically regulates the cell shape, length, and mechanical strength by hydrolyzing excess of phosphatidyl choline and phosphatidylethanamine to produce more amount of lysophospholipid lipids. These lipids are very important for crosstalk between the cellulose biosynthesis and the phospholipid mechanism. So but still there is a lot of information gap. How these lysophospholipases, lysophospholipids are directly or indirectly involved in cellulose biosynthesis. If the plant needs more strength, so PLA3 is activated. This activated PLA3 is going to break down the phosphatidylcholine to produce more amount of lysophospholipids. This lysophospholipids triggers the cellulose biosynthesis. It produces more amount of cellulose that is going to give the cell shape, cell length, and the mechanical strength to the plant. But the exact crosstalk is still not known. In another classical study done by Kim et al. In plant cell in 2019, the phosphoric acid levels play a very crucial role in regulating in regulating various plant growth, particularly in target genes. When you have a more amount of phosphoric acids, there we could be able to see the target where we have a more amount of phosphoric acid. It's going to block the clock regulators. These clock regulators are involved in lipid metabolism. So there are like this, many examples are there. Zillian and et al. They have identified SDP1 and TGT1. They are mainly involved in partitioning the fatty acids in ER membrane or beta oxidation pathway. And during heat stress, we could be able to see the more amount of more expression of HIL1. HIL1s are nothing but patatin-like structures. These patatin-like proteins are mainly involved in hydrolyzing monogalactosyl diacylglycerols to produce more amount of fatty acids. If you have more amount of fatty acid, that's going to synthesize more of triacylglycerol and phosphatidylcholine for the membrane biosynthesis. And is also involved in various ABA signaling mechanisms. Like if you have more signal of ABA, that's going to activate LPD. LPD is phospholipase D is going to activate a factor called Rho factor, binds to that Rho that responds to the ROS signals. And during hormonal stress conditions, PLDs are going to synthesize PA. These PAs are physically interact with ABA inhibitors, so that promotes it, stomatal closing and opening systems. And these ABAs are very important. This ABA signaling is very important. When ABA signaling is there in the plant, this phospholipase D1 gets activated. It's going to synthesize more of ABA. The ABI1 is abscisic acid inhibitors. When there is abscisic acid inhibitor, when there is an abscisic acid signaling, if there is no PA, so the signals will not transmit. The bond form of ABI1 and PA is going to be active, and that signals. So the bond form of ABI1 with PA, that's going to inhibit the ABI1 activity. Thus, ABA signals are transmitted to the nucleus. And it is also involved in PAP regulations. And during abiotic and biotic stress, it is very important during abiotic and biotic stress, more of PLDs are involved that leads to early senses. And also that leads to loss of crop productivity. On the other hand, people have identified that there is an lipid species called LPE. When there is a high amount of LPE that is going to block early senescence, and also stops the loss of productivity. LPE act as anti-senescence lipid molecules, and many research teams are working on developing LPE-based formulations to prevent early senescence in the plants, thereby avoiding crop damage by external factors. So with this information, I would like to conclude that the lipids are mainly involved in regulating sterile pathway. Lipid function is act as a clock regulators mediated plant growth and development. And lipids mediates the inhibition of early senescence of plants. These lipids are crucial, play a very crucial role in ABA responses, and also play a vital role in stomatal opening and closing mechanism. The fatty acid derived from the betatin-like proteins prevent the heat stress. Finally, lipids is essential for cell survival. And I thank all the organizers for giving me this opportunity, particularly Dr. Kumar sir. Thank you so much for your kind patience and listening.
Thank you, sir. Here is a question, sir. Is it possible to use these lipids in soap industry? If yes, at what extent to compare to traditional lipids?
Can you repeat the question now?
Okay, sir. Is it possible to use these lipids in soap industry? If yes, at what extent to compare to traditional lipids?
I'm not aware of the using of lipids in soap industries, but we are mainly into the how lipids are involved in cell signaling. Maybe people are working on it, but I'm not sure about it.
Okay, sir. Thank you, sir. Now I request Professor Gopalam Advirao sir to thank the speaker.
Yeah, okay. Once again, I take this opportunity to thank Dr. Venkata Rao for giving a wonderful lecture. Thank you very much, sir, for giving your valuable time. On behalf of Biochemistry Department, on my own behalf, on my behalf, I thank you for giving good lecture. Thank you. Thank you, Ananda.
Thank you, sir. Thank you.
Okay, thank you all. As per the schedule, now it is time for lunch break. I request all the participants to rejoin at 1:50 p.m. using same links for remaining sessions. Thank you all. Good afternoon, all. Welcome you all for the continued session of today's webinar. Now I request Dr. Santosh Kumar sir to introduce a resource person of this session. That is Dr. VENKATESHWAR Reddy M, Manager R&D Biological E Limited, Hyderabad, on topic downstream processing of vaccine candidates.
Good afternoon to all. I welcome you all once again for this one-day webinar session, afternoon session. So there are two sessions in afternoon sessions. So first one, Dr. VENKATESHWAR Reddy M. So it's my immense pleasure to introduce Dr. VENKATESHWAR Reddy M. Is a managing director, manager, downstream process, research and development, Biological E Limited, Hyderabad, Telangana. He obtained his master degree from Bangalore University and PhD degree from Sri Krishna Deva Raya University, Anandpur. So Dr. VENKATESHWAR Reddy also worked as a lecturer in Prakasham district, Veera Brahmendra Swami College, Prakasham district, Andhra Pradesh. He also worked as a research associate in research and development at Sudarshan Biotechnology Limited, Hyderabad. So VENKATESHWAR Reddy also joined as a senior research fellow at Department of Animal Sciences, School of Biological Sciences, Central University of Hyderabad. So he served as an assistant manager in vaccine development R&D division, Biological Limited, Hyderabad. Presently, he is working as a manager at vaccine R&D Biological division, Biological So Dr. VENKATESHWAR Reddy is a patent on novel purification process involved in planning, execution, downstream activities, and data analysis of purification process development. He also acquired good analytical skills and analytical knowledge and troubleshooting abilities. He has a vast experience in purification of proteins, especially recombinant proteins from E. coli, cross-reactive material from wild strain, and HPV viral-like proteins. He has been involved in developing alcohol-free chromatographic purification process for capsular polysaccharide purification from Streptococcus pneumonia. He is also involved in application of DOE experimentations and in purification processes. He has expertise in writing technical reports for the development of purification processes and troubleshooting downstream activities while processing large-scale production up to 200 liters. So Dr. VENKATESHWAR Reddy has published significant number of research papers in high-impact national and international journals, and he also presented papers in national and international conferences and seminars. So today we are here with Dr. VENKATESHWAR Reddy is MDest us online to enlighten us on downstream processing of vaccine candidates. So I request you, sir, please, I request you to present the downstream processing of vaccine candidates seminar, and also I request you to share your screen. Hand over to you, sir.
Hello.
Yes, sir, you can continue.
Yes, wait. I am sharing the video. 1 second. Yeah. Is it?
Yes, sir, it's visible.
Yeah.
Slides are visible.
Yes. Is it okay?
Yes, sir.
Shall I maximize?
Yes, sir.
Okay.
Okay. Thank you, sir.
Thank you. Thank you, Dr. Santos, for very much lengthy introduction. Thank you. I thank you, Davangere University, for inviting me to give this lecture, small lecture on downstream processing of vaccine candidates. Good evening, all lecturers, and good evening, all students. I'm Dr. Venkateshwar Reddy, presently working as Manager R&D at Biologically Limited. Let me introduce about Biologically Limited. No, 1 second. Slides. Yeah, sorry. Let me introduce about Biologically Limited. This is this company has been started around 40-50 years ago. These are the vaccines that are product portfolio of the Biologically Limited. BETT, tetanus vaccine, and BE-VAC, hepatitis B vaccine, COM-BE5. This is combination of diphtheria, tetanus, pertussis, hepatitis B, Haemophilus influenza type B conjugate vaccine, and JE, Japanese encephalitis vaccine, inactivated. Then PIPVAC, that is diphtheria, tetanus, and pertussis. All these three were combined. And BE rabies vaccine, BE-RAB, measles and rubella vaccine, live attenuated vaccine. Typhi BEV, typhoid conjugate vaccine. Most of the vaccines like COM-BE5, that is pentavalent vaccine, which is being administered to the children at maybe less than 8 years age, being administered by central as well as state vaccination program of Government of India. Maybe it is in Telangana also, but I don't know about this vaccine in Karnataka region. Most of them are being supplied in India as well as World Health Organization. That means they'll all supply to other small countries also from this company. And then what about other product? The product pipeline these are pneumococcal conjugate vaccine, DEPT plus pentavalent, that is hexavalent vaccine. It is under phase 2 clinical trials, and inactivated poliomyelitis vaccine, which is also under phase 2 clinical trials. Then COVID-19 vaccine, that is named as Corbevax, which is under phase 3 clinical studies. Maybe recently you might have seen that was published around 90% efficacy is there for that vaccine. It was published in the newspaper. Maybe soon will be coming into the market. Then hepatitis A vaccine, which is also in phase 3, and measles vaccine, which is in PCT studies. What this is under products under pipelines. Okay, then coming to the next structure, can you all remind what is the structure that is presented on this? This is beautiful structure of the most pandemic coronavirus. How it looks. This is 3D view diagram of that coronavirus. Coming to my topic, vaccine candidate purification. What is vaccine? First, a vaccine is biological preparation that provides active acquired immunity to particular infectious disease. That means whenever there is any vaccine is, if you administered into the body, antibody will be produced. This is the main logic behind the vaccine. There are number of types of vaccines that are available. That is inactivated vaccines, live attenuated vaccines, recombinant vaccines. These inactivated, live attenuated vaccines, it is old methodology. That means usually the virus, whole virus being inactivated or chemically treated to decrease its disease causing nature of the organism. Then these are introduced into the body. Whenever these are introduced into the body, they will body recognizes them as antigens, and antibodies will be produced, which will give us protection against that particular disease in future. This inactivated method is being used for Covaxin. This is the method that they have used for Covaxin COVID-19 vaccine by Bharat Biotech Limited. Then recombinant vaccines, after inactivated vaccine, that near new technology was recombinant vaccines. Here the protein being cloned, whatever the disease causing agent that is being cloned into the body, that is being cloned, cloned, expressed into the organism. That cloned protein being purified by various purification methods. That purified antigen or protein being administered using as vaccine, which will give immunity. Then another mostly hepatitis B virus, that is Shanvac, very famous around 10-15 years back. That Shanvac is based on recombinant protein vaccine. Then viral-like particles vaccine. Viral-like particle vaccine is a new methodology because this viral-like particles has more immunity when compared to the normal antigen. Then comes to DNA vaccines. Sorry for the little disturbance. 1 second. Then DNA, viral-like particles, these are the molecular weight of these particles will be around 4,000 to around 10,000 kDa. These are very large molecules which being recognized by the body. And phagocytosis, macrophage cells will take will recognize these VLPs, which will be further expressed on the cells that is being recognized again by the immune system to produce antibodies. Like DNA vaccines, this is also new, very new technology, and RNA vaccines. In DNA vaccines, where the gene that is coding for particular protein being introduced into the body with the help of lipid nanoparticles or something, so that they will be injected into the body. Once after this DNA enters into the body, it enters into the cell, and it uses host mechanism to produce the protein, which is expressed on the cells, recognized by the host system to produce antibodies. Like RNA vaccine this is the technology being used by whatever Serum Institute of India, Covishield vaccine being based on this technology. Here, when compared to DNA vaccines, here RNA itself is being introduced into the body with lipid nanoparticles or something, where RNA produces desired protein of interest. Then inactivated adenoviral vector vaccines, replication and non-replication. This is also another new method of in types of vaccines. Then coming to industry, how the working overview of the industry. In industry, there are 4 or 5 working overviews are there. It is divided into upstream process, then downstream process, then analytical or quality control, and blending and filling. Then finally QA, quality assurance team will be there. They will check in all these systems, on all these different processes, they will be there. Finally, they will submit whatever the sample and all. There comes the QA role, but it is also important role. In upstream process, I'll just brief other upstream process, analytical or quality control. Then my main focusing area is downstream processing. In the upstream process, what will happen is, before upstream processing development, my clone development comes. Clone development is first before upstream. Upstream is fermentation part. Coming to fermentation, before fermentation, first clone has to be developed. Clone is particular vaccine candidate has to be developed. How this vaccine candidate is being developed is, First, we'll identify what is the, suppose if you want to make vaccine for COVID, we'll usually that antigenic determinant of that virus will be taken as vaccine candidate, because whenever if it goes inside, that is being recognized. Hence, most of the time, surface proteins of the bacteria or virus will be taken, and these surface proteins are being cloned. Mainly, epitopic region will be cloned. 2 or 3 epitopes regions might be combined and then cloned. Nowadays, these cloned are amplified, or they will synthesize the particular gene. After synthesizing the particular gene, you might be knowing for gene activation, you should have some promoters and all. Translation initiation, translation termination, all these sequences should be there. Once this whole gene along with promoters, translation initiation codons, termination codons, all are there, that will go for a particular expression system. Why this expression system is where we have to express either in bacteria or yeast or insect cell line systems, because expression system is very much essential. This expression system, for example, it should be if the protein requires post-translation modification like phosphorylation, acetylation, or some others, it should be cloned in eukaryotic organism. Eukaryotes, because in prokaryotic there is no post-translation modification. Like that, based on the requirement, the expression system should be considered. Once the expression system is considered, the particular gene along with the vector is being introduced into the expression system. Then confirmation of the clone by restriction digestion and PCR amplification and all. Once the confirmation is done, then it is expressed under laboratory conditions, conical flask under around 500 ml or like that. This and they'll lyse the cells in small sonicator and all, and check the expression of the protein, and by checking the Western blot. Western blot, it probed with particular antibodies that are available already. Once if that expression is there, then they'll partially purify the protein and go for N-terminal sequencing, because we have to confirm whether the particular protein produced is ours. Even though molecular weight and all coincides, there may be some chances are there. If the protein, if there is one amino acid sequence difference is there in the protein expressed, that will cause a lot of problems in future. That means in next stage. Hence, this N-terminal sequencing will be done, and they will check after N-terminal sequencing, they will check whether protein matches with our whatever synthesized protein or not. Once this confirmation is complete, then the clone will comes to fermentation, USP, that is upstream processing. Here the process usually it will be around 3 liter scale or 5 liter scale at R&D, because at R&D initially it is too much. If it is larger volumes, it will be waste for because it has to be confirmed first. Then further usage will be purification will be done by downstream. At 5 liter scale, they will try to improve the expression of the protein by changing conditions like dissolved oxygen, pH of the fermenter, and agitator RPM to increase the growth, and the nutrient elements. All these different factors they'll consider, and they'll improve the expression of the particular protein. And once they increase the expression, they'll check for consistency. Consistency means, suppose if one batch, the the expression is around 10% or 2% of the total proteins. In another batch, it should not be less than 1% or 0.5%, or it should not be 10%. Because whatever it should be around 10 plus or minus 5 to 10% of the expression. This consistency is needed while moving on to the next level. In industry, in any fermentation step, in any step, that this consistency is very much essential. Next, once this expression is checked, the fermentation batch, they will hand over the sample to downstream processing. Before handing over, the sample should be harvested. Sample harvested usually, this is also known as clarification. That means either we have to separate the supernatant and pellet separately. Usually this is done by using centrifugation at lab scale level, because we'll be using around 10,000 G relative centrifugal force. We can separate easily at laboratory scale. But at the higher scale, around 500 liters or 1,000 liters, this lab scale does not work. Hence, continuous centrifugates will be there. These continuous centrifuges, it looks like this. This is small centrifuge, small continuous centrifuge, where I have shown some what is this separation solid collecting bank, and the separation chamber will be there. For this, inflow will be there. Based on the clarity of the sample, flow will be checked. This flow, once around 1 liter per around 500 ml per minute, like that they will check, and if the clarity is achieved, we will continue further. If the scale increases, the capacity of the continuous centrifuge should also be increased proportionally. This is how the centrifugation works. Then microfiltration. In another alternative to the centrifugation is microfiltration. Microfiltration is filtration using 0.5 micron to 110 micron filtration. Usually hollow fiber cartridges, whatever I have mentioned, showed in the slide, the hollow fiber cartridges looks like this. It is the pore size will be 0.1 to 10 microns. Usually bacteria or yeast around 3 microns to around within this range. Hence, to take out the pellet, this microfiber cartridge, hollow fiber cartridge will be used and separates pellet and supernatant. And another method to this harvesting is depth filtration. Depth filtration is also removes particle size within the particular range, around 0.2 to 2 microns, 1 to 5 microns. There are different types of depth filters are available. And let me show you once. This is depth filtration. What is depth filtration? Depth filters are variety of filters that are used of porous filtration medium to retain particles throughout the medium, rather than just on the surface of the medium. That means, as shown in the pictogram, the particles will be present within the tortuous path. The path will be in different directions, so that particle will be settled here and around. The output, particular throughput of the sample will be high. Hence, these depth filters are used. These are made using diatomaceous cellulose fibers like that. Pore size will be in range. Whereas compared to normal filtration, surface filtration, the depth filtration have ranges. In absolute filtration or surface filtration, where the particles are settled only on the surface, as shown in the left image. On the screen, these particles are settled. Cake formation will be fast. The membrane will be clogged within no time. Hence, for clarification, depth filters are preferred when compared to the screen absolute filtration. In the absolute filtration, there are number of sizes available, from nanometers to 0.8 micron, 0.1 micron. That means for bacterial retention, 0.2 microns will be sufficient. For virus filtration, different based on size of the virus, nanometers filters should be used to reduce the virus level. This is how the size of these filters will go. Around 30%, 30-inch filter will be there. 10-inch, half-inch, 1-inch, 10-inch, 20-inch, 30-inch, like that. Again, the area of the filtration is important. The area will be rises based on the inches. This sample clarity, why this filtration is necessary? Because when we go further in purification step, if the sample is not clear, these samples are clogged immediately, like TFF or any membrane chromatography column chromatography, the material will be fouled immediately. Hence, this sample clarity is essential at this step. Usually by monitored by checking absorbance or NTU. NTU is nephelometric turbidity units. Then once the pellet clarification is done with the upstream processing, the sample comes to downstream processing. What is downstream processing? Downstream processing can be defined as a series of separation and purification activities that together can produce a protein product fit for its intended use. You might be knowing intracellular expression and extracellular expression. Suppose if that protein is intracellular, we have to lyse the cells and solubilize them. We have to lyse. After lysing, we have to check whether protein of interest is in inclusion bodies or it is in supernatant. If it is inclusion bodies, take out the inclusion bodies, solubilize and detergents or something, then refolding, and then you go for purification. If it is in the lysis supernatant, this again will go for clarification and then purification. If it is the cell is extracellular expression, here pellet is not required. This extracellular expression will take supernatant and clarify and then go for purification. Now, what are the next purification techniques? Once we have received quite good amount of supernatant from upstream processing, suppose if it is around 2 liters or 3 liters, then there is no problem, we can continue. In the scale up, with increasing scale, scaling up, will be receive around 500 liters or 1000 liters. At this step, it is not possible to handle. Hence, most of the times it will be concentrated and diafiltrated. This is usually done at the lab scale. It is by dialysis membrane or centrifugation devices because of the small volumes. Suppose you will be for larger scale, usually it is done by using tangential flow filtration. This is method of best method using purification of biomolecules. Why we take TFF? For removal of host cell coloring impurities, then to keep the sample in the required buffer for removing low molecular weight impurities. For this, we have to go for concentration and diafiltration. What are the steps required for, what are the precautions we need to take for tangential flow filtration? There are different types of membrane cutoff membranes, like molecular weight cutoff membranes. What is this molecular weight cutoff membrane is? Suppose the protein, based on the length of the DNA, it will produce a protein of interest. It will be around 30 kD. This kD depends upon amino acids which are produced by the gene. For one amino acid, 110 Dalton's molecular weight will come. Based on this approximate, around 30 kD proteins are, BSA is 66 kD protein, like lysozyme is 14 kD protein. Based on that molecular weight, we have to use molecular weight membrane. For suppose an ultrafiltration filter with a nominal molecular weight cutoff, that is 3x to 5x less than the molecular weight of target molecule, is generally recommended. Because if your protein of interest is around 60 kD, you should use around 10 kD or 30 kD. Again, it is trial and error. If it is not observed in the permeate using 10 kD, then you go for higher molecular weight, 30 kD. If it is observed in the permeate, then come back to 10 kD. Like that, you have to choose. What are the conditions we should be remind? But field flow rate, TMP. TMP is transmembrane pressure, that is pressure exerted on the membrane in the flow. It is usually calculated from field pressure plus retentate pressure by 2 minus permeate pressure. It will give TMP. For each membrane, the TMP should be around, for normal membranes, around 1 to 1.5 bar. You should not go beyond 1 to 1.5 bar TMP. That membrane area and parameters for scaling up of these 3 parameters should be in, we should remind when we go for scaling up. Next, coming to how TFF works. First, whatever the image we have seen, here in the dead end filtration, the force, whatever pressure flow is perpendicular to the membrane, whatever black one is membrane. Hence, the particles are settled on the top of the membrane, blocking the membrane. Whereas in cross-flow filtration, the flow will be tangent to the membrane, where the cake formation, because of its flow, the cake formation won't be, the cake formation won't be allowed here because of the flow. Hence, feed flow should be maintained. Whatever permeates comes out will be collected in the permeate. Then, hence, this feed flow is very much essential in cross-flow filtration. These are different types of membranes that are being used. Right corner one, that is 50 centimeter square membrane. This is for lab scale areas. 50 centimeter, usually it is 1 meter square membrane area will be based on meter square. For 1 meter square, it is 10,000 centimeter square. In that, the membrane is 50 centimeter square. Here there are 3 types of membranes. Top one is 0.1 meter square, 0.5 meter square, and 2 meter square membrane. If the volumes are more, then we will go for higher membrane area. In the left panel, it is I have shown some image of TFF system. Whatever feed pump, this is feed pump. It will flow. It will pumps the feed line, either buffer or sample at particular flow rate. The pressure that is connected to feed line is feed pressure. Here is retentate pressure. Here, whatever right side it is mentioned is permeate. Usually, most of the times, permeate pressure is not mentioned in, not kept in normal flow filtration. Whereas for microfiltration, permeate pressure is required. This is how once the sample comes entered to the membrane. Here, the membrane will be like this. After the permeate, whatever comes through the membrane passes through permeate, the retentate recirculates in the feed line also. This is retentate. Finally, we collect the retentate by recirculating based on the volume of the, based on membrane area. Volume is recirculated. Whatever membrane that is attached to the whatever protein that it attaches to the membrane will be recirculated and taken into the retentate. That is how we will prevent the loss of the protein that is attached to the membrane. Then, based on this, we have come to how to concentrate. That means whatever 500 liters or 2000 liters that we got, it is concentrated based on the concentration. Around 1 mg. Suppose if you want 10 mg per ml, also we can concentrate based on the requirement. Once we have concentrated, now what we have to do? What is our requirement of purification? Based on your requirement, there are suppose if you want to immunization around moderate purified antigen is sufficient. For functional studies, around high purity 90 to 95. For therapeutic proteins and monoclonal antibodies, greater than 99% purity is necessary. Based on our application requirement, different purity were given here. Suppose in vaccines, around greater than 95% purity is required. Based on this, we will go for purification of the vaccine, around vaccine or protein, either protein or polysaccharide or conjugate, anything. Now, this thing is chromatography. Chromatography is, as you know, you might have studied in the theoretical classes, process of separation of mixture of protein or compounds, that is called chromatography. As there are different types of chromatography, you might be knowing, like ion exchange chromatography, gel filtration chromatography, hydrophobic interaction chromatography, and what else? That's it. Multimodal chromatography, new resins also observed. Affinity chromatography. These chromatography mainly depends on the functional group of the resin. Usually, in all the cases, the beads will be similar, either agarose beads or methyl acrylamide beads or silica beads. All these beads, whatever they will take, they will code particular functional groups. Suppose if it is ion exchange, they will code either cation, carboxymethyl cellulose, sulfoxymethyl cellulose, like those functional groups. For anion exchange resins, DEAE. diethyl ethyl quaternary amino groups. Based on that, these functional groups are quoted and will be receiving these ion exchange resins. Anyway, we can't do that coating and all. We have to buy resins from companies like Biorad or GE, Cytiva now, like that we have to buy. Once we have taken particular resins, mainly we will see the ion exchange chromatography. Here, the as you might be knowing the PA of the protein. What is PA? Isoelectric point of the protein. This isoelectric point of the protein is based on the charge of the protein. If the pH of the protein and the PA of the pH of the buffer and the pH of the PA of the protein is same, then the protein does not have any charge. That means the protein will precipitate. If the pH of the buffer is greater than PA of the protein has negative charge, where we have to go for anion exchange chromatography. If the pH of the buffer is less than PA of the protein has positive, we have to go to cation exchange chromatography. And here in ion exchange chromatography, the purification is based on any one of the interaction. Ion exchange, here ion. In GFC, it is size matters. In hydrophobicity, where the molecule, the functional group is like benzene groups or butyl groups, phenyl groups, like that. These have hydrophobicity. For hydrophobicity, sample should be added with salt, so that protein, it will expose some hydrophobic patches of the protein. Then another chromatography is multimodal chromatography resins. These are MMC, Capto Adhere, Capto and Capto Q resins by Cytiva. Here more than one interaction will be using in the purification, like gel filtration, size and ion. Both the interactions are used in the resin for easing the process of purification. Like with these using, these are the main basic principles while going for the chromatography, either ion exchange, affinity chromatography or multimodal chromatography. Now, before taking particular resin, even though even ion exchange chromatography, either strong ion exchange are there. What are the, now we have to select resin characteristics. Resin characteristics is how much protein is binding, how much resin is binding capacity, pressure limits of the resin. Usually at higher scale, pressures are very, very important, because the resins can't withstand pressure. Agarose based resins only, they can withstand 3 bars. Whereas silica based resins, they will withstand more than around 5 to 10 bar also coming nowadays. These pressure limits are very essential. For glass columns and all, the pressure will be around 3 bar only. Hence, the pressure limit we should be keep in mind this limitation. Hence, it is necessary to take care of these characteristics of the resin. And leachables and extractables established by the company provider. They will establish. If you buy from known companies, they will tell that we don't have any leachables and extractables. That is sufficient for us. Hence, we'll go most of the times. Otherwise, we have to give the data. The number of CIP cycle. CIP is clean in place. Once after how many cycles it can withstand is necessary, because after these number of cycles, we have to throw the resin and use new resins. Hence, all these characteristics should be keep it in mind. Once we have taken particular resin, then column, we have to pack the column. These are the parameters we should be keep it in mind. Column volume, how much volume we have to pack, that is pi r square h. Flow rate, centimeter per hour. Usually it is ml per minute. That conversion is there. I'll to show you. Column bed height for ion exchange columns, around 15 to 20 centimeter column bed height is sufficient. Whereas gel filtration, column height should be more than 101 meter. Otherwise, resolution won't be good. Slurry preparation, volume of the buffer to be added, because in small scale, you can't pack the columns in large scale. That is also important. Packing factor, this is also another factor. Once after column, by taking all these precautions, we have to pack the column. Once after packing the column, we should know whether how efficient it is. How efficiency of the column is checked by using by checking asymmetry and HEDP. Asymmetry is usually calculated by passing the column with acetone or NaCl. If you inject around 1% of the column volume, acetone should be injected. It will the peak will, as acetone shows some absorbance at 280, will be shown some peak. That asymmetry is calculated by the system itself, and manually also we can calculate. We can study. HEDP is height equivalent to theoretical plates. This varies from resin to resins. These numbers will be mentioned in the leaflet. As I told, checked by passing acetone or conductivity solution. VF for volumetric flow rate equal to linear flow rate, centimeter per hour by 60 into column cross sectional area. This is how this is required for calculation for what is that scaling up of the issue, because at lower scale also, if you measure the flow in centimeter per hour, at higher scale also will be measuring the centimeter per hour. These are necessary. Then what comes is what are the performance parameters should be considered when planning each purification step. These are the parameters we have to keep it in mind. Capacity, amount of the sample loaded, because onto the column, which is essential in GFC, because in GFC we can't load more than 2% of the column volume, that is 2 ml per 100 ml column, whereas in ion exchange that limitation is not there. Speed, how essential at the initial purification step. Speed is, suppose if we have not concentrated a sample, almost around 1000 liters of the sample if you receive, we can't keep on loading the sample for hours together. Hence that concentration is necessary. All these steps this is also essential at initial stage. Hence concentration is necessary. Then recovery, how much protein once after purification step, what is the percentage of recovery at each step. It is also very much necessary, because if you lose some of the around 70% of the protein and taking out only 10% of the impurity, then that step is not good. Hence we have to be the recovery around based on the impurity profile, we should calculate the recovery and then proceed further. Resolution depends on selectivity and efficacy. This step is very much important at the final stage of the purification. Now then next comes purification strategies. There are 3-step purification strategies. First is capturing, intermediate purification, and polishing. Capturing is because volume will be more at the purification. Mostly ion exchange people will go for capturing. Hence it was mentioned only capturing step. For others, this polishing are not necessary if the purity is not required. Sometimes you will get 90% purity here. Hence there is no need to go to other steps. Intermediate purification, usually it is HIC most of the times. Final purification is gel filtration. It again all depends on your protein of interest. This is how sometimes sometimes the hydrophobic interaction people will keep it as initial step followed by EX, finally gel filtration. In all these cases, gel filtration it will comes polishing step only, because it is volume will be high, lower we can concentrate. At this step it is very difficult to reduce the volume at that level. Next, suppose first what we have to, all these parameters we have to see before loading onto the column. This after loading the column, usually the sample should be whatever equilibration. First we have to clean the column thoroughly, then pass 10 to 20 CVs of the column, so the pH and conductivity of the sample should reach the sample. Our sample column pH conductivity should be reached the sample pH conductivity. Once after reaching, then sample application, then unbound molecules are removed. Unbound molecules are taken out by passing 2 to 3 CVs of the buffer. Then gradient elution, usually around 10 to 20 CVs will be kept, because we don't know at what particular CV the protein of interest our protein of interest will come, and then high salt wash is given, then re-equilibration. Based on this profile, we'll check all the fractions on the SDS page, and we know SDS page. After checking and establishing the protein of interest is there, suppose if it is around eluting at the middle of the gradient at particular around 100 millimolar or 200 millimolar step gradient we can keep. Later we can further increase the purity by giving again linear gradient layer. Like the initial step should be this way. After once, if you have at particular, suppose you thought that your protein of interest is there at this step, at this final, the different step gradients are given so that the impurity proteins come out, the protein of interest will come at high salt, or at around to in between. If it comes out, first one gradient should be given, followed by the required gradient. Like this, the protein should be purified. Now the comes to the sample. In all the again parameters I have mentioned, it might be a little bit repetition of the previous. Feed sample parameter should be consistent. The pH, conductivity, protein concentration, whatever we have loaded, it should be consistent. Sample consistency, the batch variation should not be greater than 15%. Sample retention time, while application of the sample, minimum 6 minutes in the ion exchange chromatography should be mentioned. Again, it varies from protein to protein. For maps and all, initial purification, so much retention time they will keep because the volume will be higher. At this, these are parameters should be kept in mind. Now, suppose I have shown one of the image of different stages of purification. Here, first one is molecular weight marker, where the protein of interest is 10, 14, 25, 35, around 45 kD protein is there. This is lysate. After depth filtration, third lane is, fourth lane is after concentration of the sample, the fifth lane is chrome 1 pool. Again, some lower kD protein and all there. Here also on protein was there. Hence again second column went. Then after second this is concentrate, bulk is 0.2 micron filtered. This is how the pure molecule should be. There are no other impurity band should be seen on the Western blot, or protein or silver staining. Again, purity will be checked. Next, how confirmation of the purity. For suppose the protein what we have purified like previous gel, whether this protein is pure or not, we have to check. Usually it is done by densitometric analysis, checking the activity of the protein by ELISA, enzyme linked immunosorbent assay, or structural determination by CD. CD is CD spectroscopy. Sorry, somehow I forgot that. Then NMR spectra, nuclear magnetic resonance spectra, or aggregates are checked by using HPLC. This is the gel I have shown you. On the gel you can see for reduced it is BME, beta mercaptoethanol is added to reduce the disulfide bonds, where the protein will move little higher at higher molecular weight when compared to non-reduced protein, where the protein moves, because if the protein disulfide bonds will be there, it is in globular in shape, hence it will move little faster. Whereas in because of in reduced condition, because of reduction of disulfide bonds, linear molecules will be formed, hence migration will be slower. This is how we can check on whether it is the protein correctly folded or not. It can be checked. There is a slight difference in the migration pattern on SDS based gel. Okay. Again, this is the gel of densitometric analysis. Here we have in the second lane I have shown some partially purified protein. We'll check. It is a software by Cytiva instruments. we have to see, select all the bands that are present. Then the system itself view what is the percentage of the purity of each band. Here, first band it is 87%, second band 4.9%, and this is 7.19%. On one more thing I can show you here, what about not mentioned in the lane. These are non-reduced fractions of these two, where the protein is mostly seen on the well. This is of VLP, viral-like proteins, because of very high molecular weight, around 20 to 30,000 kDa. It is not come down from the well. Whereas this third lane impurity could be seen here. Another one more impurity it is seen here, where a protein of interest not migrated because of very high molecular, which is VLP. Next, analytical development. That's where the giving pure product is a downstream processing requirement. We have in downstream we'll give the pure protein or polysaccharide or conjugate. What comes the analytical is whatever the in process steps to check whether whatever sample we have received from the upstream is correct, or whatever sample after chrome 1 sample or chrome 2 samples, whether our recovery is similar to that of previous batches. We'll collect some of the intermediate samples from these batches. These intermediate samples will be sent to quality control for checking purity or concentration or whatever we have mentioned in process. A few critical points, the yields are checked before proceeding to the next purification step. These are called critical control points, because we have to keep some points. Otherwise, based on the concentration that we received from the upstream, we'll keep some particular volumes. At downstream, in the production, it is not possible to change the volumes or concentrations. The volume slightly can change, but the concentration should not be varied. If it is greater than 8 mg means it should be. It may go up to 10 or 11, but it should not be less than 8. If that volume minimum concentration is not attained at the intermediate stage, then 8 mg concentration may not come at final stage. Hence, these critical points are critical control points are essential to get the concentration. Hence, these are very much necessary. Once all these samples are analyzed by quality control. Once we have purified that bulk is 0.2 micron filter, we will send it for COA, that is certificate of analysis. Usually it will be pH, concentration, protein impurity, test of chemicals we have used in the in-process, endotoxins, bioburden, sterility. All these, for every batch they will check all these. This is the specifications, appearance. Specification they will mention method and particular results of the batch, that is mentioned in the 4th line, I believe. Okay, these are appearance, total content, protein content, particle volume, average molecular size. All these are endotoxin. Endotoxin, what is less than 10 European units per microgram of protein. Like that, for every step, what is the value? pH should be 7.2 plus or minus 1. Like that, it should be within that range. Then you see total protein content is greater than or equal to 25. Here 176. PS content is greater than 50. It should not be less than that. These are all called specifications which will be monitored by in-process QA and all. This is how quality control. There is only one work. They'll just receive the sample. They will test the samples. Like that, other vaccine candidates that are being purified is polysaccharides, that is alcohol-based purification, chromatography-based purification, detergent treatment. That like conjugate, polysaccharide conjugate vaccines, conjugated to proteins also, they will purify by TFF and gel filtration chromatography, and viral-like proteins also. Because of their large size, usually different resins from conventional are used in purification. These are different vaccine candidates being purified in our laboratory also. Next comes to 1 second. And one more thing I just wanted to, just in the overview initial slide, I told you different, what is that, production, upstream process, downstream process, analytical, blending and filling, whatever. Once we have received the protein of interest, quality control, that protein passes all quality parameters, then it will go to blending and filling. There where they will add adjuvants like polysorbate or adjuvant fronts, complete adjuvant and all they will add, and then again go for filling. All these in process upstream, downstream will be monitored by QA. This QA process, they will check, observe each and every point, every stage. They will be there with us and check whether we are doing correct, whatever they have mentioned written in the BPR. BPR is batch processing record. They will check and they will make sure that we are following BPR. Like that, each you should be cautious, or there is no other option. Sometimes if it is upstream means you should be, your career will be mostly in the upstream part, or if you go to purification, it will be in the downstream part. Most of the time it won't come chance to move from one part to upstream part or downstream part. If it is analytical also, mostly it will be in analytical or quality control testing, and blending and filling also. Mostly they will be confined to particular areas, because whatever upstream person, he can handle what is fermenter. We can't, this blending and filling guy can't come and handle this fermenter. Like that, whatever, these are different departments that are being categorized in the industries where we'll work independently in those departments. Okay, you can ask me if you have any other questions. Thank you for being patient with me. Okay.
Hello.
Yes.
Reddy sir.
Yes.
Hello.
Yes, sir.
Yeah. So the purification of this, what you say, vaccine candidates. So far you explained regarding the traditional and conventional methods. Okay.
Yeah.
So is there any alternative methods? These are the very lengthy processes. If you want to get a pure protein, it will take some more time.
Yes.
So that's what. Is there any alternative research is going on for the single step purification of these kind of candidates? See, if it is affecting very quickly, then it is very easy to control, like pandemic type of situations.
Yes.
Is there any such kind of research is going on? Is there? Please explain.
Actually, this is whatever we have done because of this pandemic. These mostly mRNA vaccines that are coming, no, because there is no need of purification of proteins, and only at the initial stage, mRNA, if they synthesize, they will produce that mRNA in large scale, and they will just mix with the lipid particles and they will inject. That is very conventional and very fast compared to this conventional method, or mRNA, or this DNA vaccines also. This DNA vaccine also, instead of mRNA, they will directly inject that DNA along with only DNA will be given. This is very short method, and compared to, and another short method is whatever Covaxin it is following inactivation of that whole virus. That is conventional one.
Okay. Sir, one more question regarding COVID-19 vaccines are available in the market and the government are supplying, no?
Yes.
So these vaccines, they are earlier generated in the first wave of COVID, that is in the month of March or something, 2020. So now day by day, the what is COVID-19 is changing its strain, and genetically it is modifying. So
yes.
Still, they are giving same vaccines only for the public. So, in that context, at what extent that vaccine is sustainable? That vaccine is fight with new variants of, say, in October, there is coming one more wave, that is third wave. Is it workable for that wave?
Hope for the best. That's it. Because a virus, it is natural ability of the virus to mutate itself. Because they are more, we can say, advanced, like bacteria and viruses, they have the ability to mutation, whereas humans, we don't have any mutation. That is their natural ability to mutate. We can't stop that virus to mutate. because of its survival, it is it is mutating. Hence, it is suppose if you take antibiotic, if you are using continuously, the bacteria it will acquire that antibiotic resistant genes from nature by only few sequences, only flanking sequences. If by taking those flanking sequences, it will acquire the whole cassette of that antibiotic. Hence, it is very dangerous for the environment and for us also. Hence, that is their natural ability and to mutate. And one more thing, most of the times we may get this resistance, because all of a sudden we may not get. Exactly, we can't tell what is the mutation itself. Whatever the particular protein or spike protein we have raised antibody, if that closes completely in that mutation, if it is not exposing on the surface of the virus, it may not get, we may not get that resistance. If it is not completely immersed in its structure, if it is exposed a little bit, we may get that resistance. It's all fate. That's it. Most of the times, it's not in our hands. Hence, we should be careful.
That's what. Already we are having some Covaxin and Covishield right now, but the virus is continuously mutating. Then there is a fear of that. Whether this vaccine is working on that strains or not. That is the major question right now.
Yes, that is dangerous question. We can't do anything. Hence, HIV for few viruses, they are not able to generate these vaccines itself. There is no other, they are not able to predict that. And that too, this is at least people, we are thinking that at least we will get some immunity so that we will fight against that pandemic. That's it. Hence, we are using this.
Sir, is there any possibility of present existing vaccine is modification for new strains? Any methods are available for that, like that?
Any method is nothing. Nothing means
likewise some modifications in the vaccines, and that can be fight with any new variant.
Yes, they are trying. They are trying to expose. They are collecting whatever the data they are sequencing. Now it will be in public, and they are trying on the new virus, new mutants also. They will also try, but it will take time. It's not new. It's very far. It's not very fast generation. No, it will take.
Yeah, getting vaccine is a very tedious process.
Yes.
Yeah. Okay, sir. Thank you. Nice presentation.
Yes. Thank you.
Thank you, sir. It's a very informative session. Here are a few questions.
Yeah, sure.
First one, can mRNA-based vaccines can change my DNA?
mRNA-based vaccines, I don't think so. It won't change. Whatever the mRNA, it will only use the resources present in the host cell. It is modified in such a way that it won't go and bind to the DNA and all. It is just to use host system and produces its own protein that is necessary for that.
Okay,
there are no evidence, I think, up to now.
Okay. And another question is there any information about the herbal medicine given in Nellore for COVID patients?
No. Not. It is not yet given out. No, whatever they have studied it will improve our natural way of improving our immunity when compared to the. It is immunity. We are improving our immunity, thinking that by taking those things we will get some immunity so that we won't get affected by the pandemic. That's it.
Okay, sir. One more question. Last question. How complex is multivalent vaccine synthesis?
Yeah, that is a good question. Actually, it's not much complex. Actually, whatever most efficient epitope will be taken now. Maybe people are trying whatever our Corbevax biologically is taking recombinant binding domain. Maybe to combine 2 to 3 complexes, they'll put together and produce the protein. It's not much complex.
Okay, sir. Thank you, sir.
Thank you.
Now I request Dr. Santosh Kumar, sir, to thank the speaker.
I take this opportunity to thank Dr. Venkateshwar Reddy. Thank you very much, sir. Thank you for sharing knowledge and sharing your experience. Thank you, sir, and also thanks for spending your valuable time with us. Thank you, sir.
Yeah, thank you. Thank you.
Now I request Dr. Pramod SN sir to introduce speaker of this session, that is Dr. SOMASHEKARA N, co-founder and CSO All in Life Science Private Limited, Chennai, who is going to speak on the topic my experience so far with special reference to nutraceutical industry.
Okay. Well, good evening, everyone. Myself, Dr. Pramod. It is a great pleasure to introduce the speaker of the presentation, Dr. SOMASHEKARA Nirvana Shetty. He is the co-founder and CSO of All in Life Sciences Limited, which is located in Chennai. Dr. SOMASHEKARA holds a postgraduate degree in biochemistry from the Department of Biochemistry of then Kuvempu University PG Center, and which is presently at Downgrade University. He is the alumni of the department. Later, after his post-graduation, he obtained his PhD degree from Swinburne University of Technology, Australia, in joint collaboration with IIT Madras, India. Dr. Chemist with extensive research and development, primarily in innovation and development technologies. Prior to OLN, Dr. SOMASHEKARA served on various research positions in pharmaceutical, nutraceutical, and agri-food companies, with main responsibility being new technology development. Recently, their Life Sciences Private Limited has been awarded National Award in 2021 by Technology Development Board, Department of Science and Technology, that we call it as DST, Government of India. And thus for the development of nutraceutical product called Ginfort, which is a 27% concentrate of ginger extract. Today, Dr. SOMASHEKARA is online with us to share his journey. He titled his talk as My Experience So Far with Special Reference Nutraceutical Industry.
Thank you, Dr. Santosh. Your voice was breaking, but still, thank you very much for the nice introduction. So I'm just going to share my screen. Give me a minute. I hope everybody can see my screen now. I hope I'm everybody.
Yeah.
Okay, so today my presentation is actually slightly different than what you heard so far. So let me just tell my name. My name is Dr. SOMASHEKARA, and I'm the co-founder and CSO, Chief Scientific Officer of All in Life Sciences Private Limited, a nutraceutical company which is there in Chennai. So in another 30 minutes, what I'll do is I'm going to share my experience as an employee, as an entrepreneur, till I received the national award recently. So thinking that this will be helpful for the students to plan their career well. So first I'll just talk about my experience as an employee till the national award. And then I'll briefly talk about the All in Life Sciences, the growth of All in Life Sciences, and the opportunities in nutraceuticals, and then how the process of product development in nutraceuticals. First, let me start with my journey as an employee, the national award. Here, the main objective of the presentation is, as I said, it's entirely different than the previous talks. It's one should learn from other experience and do better. See, why I put this statement here is like, if you want to learn from, especially for the students who are going to pass out and look for the careers, start the career. So if you have to learn from your experience, and it's going to be like very expensive and consuming, time consuming. So when you have a resources, like my suggestion is like you talk to your seniors, the people in academic institutes, and also the industries, and take their guidance and proceed forward. You can plan your career well. So I'll just briefly start with my schooling. So like if you have like the kind of any questions in between, or I think you can stop me and ask. Otherwise, like I'm open to the question at the end of the presentation. So I did my schooling in a village called Muktihalli, which is very close to Chikmagalur. It's a government school. Then I did my PU and degree in government college in Chikmagalur. After that, I was lucky to get the admission in PG Center, Kumpu University, where you all staying now. Earlier it was under Kumpu University. I was lucky to get the admission. So there also, like I asked a couple of people which degrees, which PG degree is good in terms of like future. So like I applied even for the chemistry and the botany and zoology. Then later people have told that, like few of my seniors told that the biochemistry is good. That worked out to be good for me. So I passed out my PG degree in 2000, 1999 and 2000. The first thing which comes to the mind after the PG degree is the UGC exam. So yeah, it came to my mind as well. Then I learned from my senior, a couple of my seniors, saying that UGC exam is like very difficult to crack. So my attitude towards the UGC changed. I didn't prepare, went and wrote the exam, but like I failed. I didn't go through the exam. So I hear like the 1 point note is I didn't bother to talk to the people who passed the UGC. I just taken the suggestion from the people who felt that like UGC exam is very difficult. Then it so happened that like parallelly I wrote another exam called BAT, Baba Atomic Research Center exam. So like this time, like I just, yeah, probably because of like one of my classmates who was smart and intelligent, then I got inspired to write this exam and I got through this exam. So after going through the exam, like I got a PhD admission in Central University of Hyderabad. So I'll tell you like what has happened here. Like it's very interesting. So I went there and I completed all my admission process. Then like I went to the department. It was in cellular and molecular biology department. I came to know that there's no scholarship for me because I didn't go through, I didn't come from like UGC. Only for the UGC there was a scholarship. Then immediately I took a decision saying that, okay, this is not right for me to continue the PhD without the scholarship. So then immediately I decided to quit. So here one mistake I did is like I didn't consult anybody. So once I came back, then I started actually talking to my brother. He said that he could have called me and spoken about it. I could have actually sponsored. I missed a great opportunity there. And even the Central University Administration Department, there was some like a scholarship which was not like as good as the UGC scholarship. They used to give it for the students who are not having the scholarship. Even I didn't actually bother to go to the administration department and explore. The point here is like you need to like keep your eyes open and then explore possibilities to do the things in a positive way. So then like one thing I did which like worked out very well is like instead of going back to my hometown, I stayed in Bangalore. I stayed along with one of my friend who was working in IHR, Indian Horticulture Research Institute. So here, like I just become a little bit cautious. Then I started every day. I used to actually get up and get ready at 9 o'clock, and I used to list out the institute. Initially, I thought like I'll just apply to the institutes, not to the private companies, because like I thought I'll just work in a project, get 1 or 2 years of experience, then move to the private industry. That was my idea. But here, like how to get the job. See, like there are various ways people try, but like my approach was slightly different. So then I listed out, I used to list out all the institute to which I want to visit. And then particular day, like I used to get up and then get ready at 9 o'clock and then go to the institute and go through all the department notice boards. See, what happens is many of these research positions will not be advertised in paper, the online. That time it was like the kind of online, these things was a little bit poor. So this was in like 2000, 2001. Then like one such instance is like I went to one institute called National Dairy Research Institute, which is there in the Bangalore. So then I gone through all the department notice board, and so I happened to see the dairy chemistry notice board. There was one advertisement for the research associate position. Then I like so happy. Then I saw I didn't stop there. Just like I didn't just apply and then keep quiet. So what I did is I visited. I like went and spoken to the principal investigator of the project. I went and spoke to him. I gave my resume also. Then like he said that, okay, you wait. So you may get a interview call. That time you come and attend the interview. So I waited for 1 week. Then again, I went back and asked him. I said, yeah, you have to wait. Then I went second time and third time. Third time, I don't know really like what has happened to him. He felt that I'm really interested in this project. He said, hey, you're from a biochemistry department, and this position is for the dairy chemistry department. So like you need to have some knowledge of dairy chemistry department, because there will be a big interview team who will be asking various questions on dairy chemistry. He said like, I can help you in one way. So I can give you an access to the institute library. You can go and study. Then I went there and studied for like nearly 10 days. Afterwards, I attended the interview. I gone through the interview. I got the job, that research position. So I was really lucky, because around like 20-25 people. So I was selected for that position. Later, like I just didn't keep quiet, because like I had I'd made like so many mistakes earlier while during my PhD admissions or whatever it is. Then I don't want it to repeat. Here, immediately, like after completion of 1 year in this project, I started talking to the people in private industry, because like as I said, I just wanted to join an institute to get the experience, then move to the private industry. So while during this, I got the government job. So that is in Forensic Research Laboratory, which is there in Hadugodi, Bangalore. So, but I didn't join. I thought like that is that is not the right job for me. So I need to move to the private industry. Somehow like that was my decision, my opinion. So then I didn't join that job. Then I started applying to private companies. I got a call from like one American company called Kemin Industries. It's an agri-food company. So went there, attended the interview, gone through, like got the job. I got the job as QC chemist. Here is the catch. So here, for any students, once after the PG degree or whatever it is, so you need to look at what industry you are looking for, which company and which department. It's so important, because you need to ask one question yourself. Can I see myself doing this job all the day, every day, and many years? So sometimes some jobs will be very like monotonous. So I didn't think all these things. I think this is like one of the mistakes I did, but at that time, like I was not having much of resources. Then I joined the company. And so why I'm stressing here the industry and the company and the department is, you're going to continue, like once you join the department, not always, but like in most of the time, that will become your field of career. So I joined as a QC chemist there. So I completed 1 year, and then I thought like, you see, you need to look for two things when you start your career. First, initially, like, are you learning and are you growing? So these two are very important, right, for anybody. So if there is no learning, I think that is not the right job for you to be in. So here, like QC, I think like you heard in the previous the session also, the presentation, QC job is like the product will come, you have to test as per the product spec and give the test reports. That's all you need to do. And even all the methods will be there in the department and the company, which are already like validated. So I did for 1 year. Then I started feeling that, okay, it's like very monotonous. I'm not learning anything. There was a growth because like, since I was like a bit hardworking, so I got promoted very soon. But I thought like this is not the right department for me. Then I changed the role. See here, like not many companies allow this, because when you change the role, you will be considered as a new like candidate. So you will lose whatever the experience you got. So in QA, I started a new role as the auditing person. So I used to take care of ISO and HACCP and so on, AIB, so on. Here it was much better because like I used to, I learned different things which are not like kind of a QC, just doing the test and then giving the report. And I also got an opportunity to visit various countries. See here, like this is one more advantage of being in an MNC. So this company was having a manufacturing facility in 14 different countries. So I happened to visit around like 4 or 5 countries during that time, and then got an opportunity to work with a different team of different countries. So then like I worked for nearly 4 years. Then again there, like I thought, okay, so all these things, the systems and the auditing, everything are implemented. So what next? So then I thought, okay, research and development department may be the right things, because you will be doing different things at least like every alternate year. You'll get a new roles, new projects, and you'll be able to do handle different equipments, different assays, and you'll be part of a product launch and all those things. So it was very interesting. So when I moved again, I said like I really was thankful to that company, because the president of that company allowed me to move. Some of the companies will be very, very good. So they will actually take care of the employees' growth as well. And this was like one such company. Then I moved to the R&D. So there I worked for 3 years. So I was enjoying the job. See, these are all like I've actually done in the same company. But I suggest like any newcomer not to change the company very frequently, because no company will take you. So if you are changing the job every year, particularly I've interviewed like many people from my company as well. So if you are changing the job every year, they'll think that you are not stable. So they'll not give you a job. Means they'll not prefer you. They'll prefer like somebody else who is very stable, at least for 3 to 4 years. If a person is staying in a company, that will be considered as a good time. So here, like after 3 years, then I noticed one thing. So I was actually learning a lot, but I was not growing like much in terms of career. The reason is I was not having a PhD. So the people who have a PhD in the research, particularly in the research and development team, they will be preferred for the promotions and as a group leaders and so on. So then I felt that the PhD is very, very important. And then I thought of doing the PhD. Like I spoke to the company heads and the president, but there was no option for me to do a PhD in the same company. So then I decided, actually, see, I went in search of like some kind of learning and then growth as well. So then I decided to quit the job. See, it was an MNC. It's kind of US company. If I have stayed there, I would have settled very well in terms of salary as well as the job security. But I was just looking for, then I thought like I have to do a PhD in order to grow further. Then like I started looking at the companies, and then I got a job in a very small pharma company. It was like a kind of the few months old pharma company. So here actually again, I feel like I have taken a good decision. Here, like instead of joining in your early career, instead of joining a big company, it'll be good to join a small company because you'll learn a lot. So big companies will have all the departments, so many people. And if you join the company, you will become a part of small group, and you'll be doing a small part of the whole product development or whole project, whatever it is. But whereas in the case of a small company, which is a startup, it's called as a startup. So if you join there, there'll be less number of peoples and a few departments. You will have access to all the department, and then you'll have like kind of learn a lot. You can probably learn all product development. Same thing happened with me. So in this pharma company, when I joined, it was started by ex-vice president of Sun Pharmaceutical. Sun Pharmaceutical is a big pharma company. Basically, I joined this company for the PhD, as well as like I learned a lot, because here like I got involved in all the stages of drug discovery. It was like a pharma company that was into drug discovery and development. So here, like from moving from agri-food to pharma was difficult. So what I did differently here is the president of this company used to go to many conferences and used to give a lot of talks, presentations, but he was not having a time to prepare the presentation. So what I did is I told him like, I'll prepare the presentation and give it to you, because so it was a pharma industry, it was new for me. Then I decided I'll take an extra hours and then prepare the presentation and give it to him. I used to prepare presentations for him like during the after the office hours, Saturdays, Sundays. For all his, like probably for 3 years or 4 years, I did all his presentation. I prepared the presentation case. Like the beauty of the preparing a presentation is like when you're preparing a presentation, you need to read a lot, because you cannot copy and then paste it. So while doing this, I learned a lot. I actually learned how drug discovery takes place, what are the various stages, how to handle the various teams and all those things I learned. That was really helped me a lot in order to stay in that company for quite a few years. Then I actually parallelly I applied for a PhD. Luckily, like I got a PhD position in IIT Madras. This was under a collaboration between IIT Madras and Swinburne University of Australia. So I started working on the PhD also. That is also during the Saturdays, Sundays. I used to go to IIT and then do my work. And every year, like I used to go to Australia and do the work over there also. So I completed most of my PhD work, and by then I've completed only the few part of the drug development in that company. You must be knowing like the drug discovery and development, if it is a new chemical entity, it's going to take like around 12 years to develop a product, and then around $1 billion. It's very costly affair. You cannot be like a part of the whole complete development there. So then I thought like at that stage, I was having nearly 12 years of experience. And then I decided I need to do something like unique, because I have acquired some experience, not all, probably some experience in the agri-foods and in the pharma. I was knowing how to handle the team and also how to develop the products. So, but then like I thought I need to do something different. I need to start a company. So that was like a big decision, but I thought like, okay, I should be doing that. I started working towards that, and then like thought like which fields again. So if I whether to start the company in the agri-food industry or pharma industry. If it is agri-food industry, so I thought like it's a kind of a, see, agri-food, when I say it's going to be feed related, feed supplement and all those things. And here the business is high volume business, but low margin business. You need to have a marketing muscle in order to survive there. Then I decided, okay, that's not the right thing, because my skills are like product development. Then I thought about pharma. As I said, pharma is like very expensive, costly affair. I didn't want to touch. What next? That time I was like, while I was studying, I just came across Nutraceutical, which is like upcoming. The whole world is actually moving towards nutraceuticals. Nutraceuticals is nothing but anything which is derived from the food and the natural product, which is basically for the prevention of the disease or chronic diseases or like healthy aging, so on. So, and then whole world is actually moving toward that, and then came to know that there's a good scope and this industry is going to grow like how pharma company has grown. So because everybody is like health conscious now, rather than like treating a disease a curative, so everybody is looking at the preventive measures. So then I learned about nutraceutical partly, and then quit the job. So I quit the job in 2014. Like it was like for me at that time, it was like a highly paid and good job. But of course, it's a small company. But I decided to quit and then start the company. So it was like it's one of the big risks I've taken in my life. So you need to like when you start a company, you need to plan your finances. So because like every like month you need to like pay the salaries, you need to have sufficient money, you need to have a people to develop the products, and you need to have people to market the developed products and so on. So it was like a big decision. So but I really worked on that for like 1 to 2 years, and then started the company. Officially, like I registered the company in 2015. It's named it as Volin Life Sciences Private Limited. This company is based in Chennai, and it's a nutraceutical company. So we have our R&D center as well as the manufacturing unit in Chennai. So then after this, in 2016, I got the PhD degree from the IIT Madras and then Swinburne University. The degree is actually from the Swinburne University of Australia, and the IIT Madras was like these things. So then later, so I got very recently, I got the national award for commercializing the product based on a technology, unique technology. So from the government of India. So this is under MSME categories. So this was actually the most tough period for me. The moment I quit this job, had to go through a lot of hurdles in terms of finance, in terms of managing people, keeping the people together, because the team is very important. So you need to keep them together. So if after starting, because of various reasons, if they split, your company is gone. So then we also taken a very like big step of going to different country and exhibiting and participating in an exhibition. So that is another big decision, because normally when the small companies, like when the people start a small company in India, they'll look for the business in India itself. So they'll not normally explore the international market, but I didn't do that. So here, like I would like to stress, like you'll have like a lot of opportunity when you pass out from this university. So after that, like you'll have an option of joining the academic institute, pharmaceutical companies, food industry, biotech industry, and allied industries. But you can add nutraceutical industry into this list. Currently, nutraceutical industry is a $400 billion industry globally. And in India, it's actually upcoming, it's around $3 to $4 billion industry in India. So various pharma companies are venturing into a nutraceutical company because they understood that the whole world, the population is moving towards natural product, prevention therapies, and then organic products and so on. So they know the importance of nutraceuticals in the healthcare industry. So nutraceutical also, like many countries are promoting nutraceuticals, because nutraceutical, by preventing the diseases, these are reducing the health burdens, so treatment cost. So that's why this industry is like becoming very popular globally. As I said, it's a fast growing industry. Focus more on prevention therapies and safer, because these are derived from natural products. So these are like more safer when compared to pharmaceuticals. In case of pharmaceuticals, like if a company is developing NCEs, new chemical entities, 99% of the time, so probably I would say 99.9% of the time, the product, the molecule will be toxic. That's why the drug discovery is very, very expensive. So here you don't have that kind of like problem. It's a good opportunity, like for a career. So if you want to start a career, so if you want to continue in the nutraceutical industry, since the industry is booming, so you will have a good career growth. You'll have a lot of option to grow in this field. The second thing is, as an entrepreneur, so since this is not like a very expensive, normally like we take around 3 to 5 years for the product development, and we spend around 4 to 5 crores per product to bring it to the market. So this again depends on the particular product, but this is actually, average we spend this much time, and then they around like 4 to 5 crores of rupees for developing a product. You can actually, if you want to start a company, you can start like product development company or manufacturing company or like extraction company. There are like kind of extraction and formulation. There are like different segments in nutraceutical industry. And also the farming is another opportunity. So there is like high demand for medicinal plants. Those will go for the extraction, followed by the formulation development and various applications. This is briefly about the Olin Life Sciences. So we are, as I said, like it's an intellectual property driven company. We focus more on developing a patented and clinically validated nutraceutical product. So we don't take an approach of the making the extract and selling it. We really do spend a lot of money and then develop a lot of the science around the product, validate them, and then bring it to the market. So we have 13 patents in 7 different countries. Then we have like 4 international brands, and recently we received the national award. So I'll tell you like the product development process. This I'm actually talking because, so you should not assume that like this is a very simple process. Nutraceutical industry, like nutraceutical development process is not a very simple. We follow the process which is more or less similar to the pharma, but like we are not going to take that much time, and it's not that much expensive. Probably, as I said, like we'll spend around 3 to 5 years of time in developing a product. First, how do we approach in developing a product? First, actually take, for example, our Ginfort, a ginger extract, which is like highly concentrated and only one in the world. So we gone through the products which are available in the market, and we came to know that the concentration of actives called gingerols is only 5% in the product, powdered product. Then we thought like, okay, we need to do something differently. We studied the phytochemical properties of the active molecules, the products. This is the basic information we collect. And if it is not there, we develop this, the information. Then we'll go for formulation development or technology development to address this issue. So one such, the technology we developed is Activism technology. I think like this information is there like everywhere in the internet. So based on this technology basically creates highly concentrated products. I cannot explain the technology, how it works because for the confidentiality reason. But it actually basically produces highly concentrated product. One such product is ginger product, which is 5 times concentrated compared to the industry product. Then we do preclinical safety studies. Since you have created the highly concentrated product, the international market, the consumer will not accept that as such. They'll ask the general ginger powder or the products are there in the market. We know that they are safe for consumption, but is your product safe? So to answer this, we do the safety studies as per, there is a guideline called OECD guidelines. As per that, we do a lot of safety studies. This one alone will cost around 50 to 60 lakhs, the safety studies. In case if you are addressing the problem of absorption, bioavailability, or bioefficacy, increasing the bioefficacies, and so on, then you have to do a lot more studies here, because you need to do studies in animal models, and then the safety studies, and then the efficacy studies. And after this, once we prove in the animal studies, then we'll go for a human safety studies. This is again depends on what kind of indications you're looking for. See, here in nutraceuticals, while you're marketing, you cannot make the big claims. You need to make claims called soft claims. You can say, like in case of drugs, you can say this is for the treatment of osteoarthritis, but in case of like nutraceuticals, you have to say it's for joint health, not as osteoarthritis. Although you will be doing a trial on osteoarthritis, you're not allowed to do that, because nutraceuticals are considered for like improving the health, not for the treatment. Then we market this product. Right now, like we don't have the marketing department, but what we do is, so we partner with the global companies who are having the marketing muscles to market our product. And we participate in like many global events, like exhibitions, and then we'll sell through these companies. So we are basically like manufacture and give it to them. They will sell it to big companies like Nestle and all those things, where they will include our product. I've taken Nestle just for the example. We are not selling it, but there are like other companies like Nature Bounty and all. They are using our products. So like even this is also just for the examples, because I cannot disclose our partner information here. So that's how we do our business. So this is briefly about the process of development. So this is briefly about my journey as an employee to the employer, entrepreneur, and then recently received national award. So we are planning in the future, like we are planning to have our business expanded to US, because there that is the central place where we can expand our business to the various countries. So that is what we are planning to. But like our base will be in India. So we are planning to build another manufacturing facility in Chennai, Tamil Nadu, and then cater to the global market. So thank you. So I'd like to actually thank the organizer for giving me this opportunity to share my experience. And I hope like whatever I shared now, it will be useful for the students to plan their career well. If you still need like any kind of like suggestions or help, you are always welcome to reach out to me, or you can call and talk to me. I'll be like very happy to help you. Thank you.
Thank you, sir. It's really interesting and informative session.
Hello. Hello.
Yeah, I can hear you.
Yeah, sir, can you please tell me your manpower of your company and what is and all those
like in the R&D center, so we have 22 people, and then in manufacturing, it's kind of a contract manufacturing facility where we have people of seven people for manufacturing product and sell it, means give it to our partners. Also, like when I say 7 people, it is also like supported by the pharma company, which is like supporting us for the manufacturing.
Okay, okay, sir. Okay. And for example, like Ginfort, per year, how much quantity of that product you are manufacturing and marketing every year?
I'm sorry, like I'll not be able to give this figure, because that's the problem with the private companies, but this will be in tons.
Yeah, okay, okay, sir. And one more, sir, I hope you did PhD, no, sir, and somewhere in abroad, something like that you said.
Yes, yes.
Then how it is possible, 2015 you started your company and you were doing somewhere 2017 or 18 PhD.
Yeah, yeah.
How you managed this running of company and doing of PhD?
It was really, as I said, like I started my PhD when I was in working in a previous pharma company. But like it was like a kind of separate project apart from what company was doing. So that's why like even after coming out of that, I was able to submit the thesis and get the degree. So I used to mainly work in Saturdays and Sundays. So, and then after office hours, and I used to use all my like leaves, paid leaves, for doing my research work. And
in which field you did? In which field you did PhD, sir?
It's pharmaceuticals. And also, like, let me finish. Like, I used to take around like 1 month leave from the company. I used to go to Australia, work there, and talk to all my the professors, and used to come back. So this is what I used to do. Okay. Yeah, sorry. Like the field is like pharmaceuticals. I worked on Alzheimer's disease.
Okay. Okay. Thank you.
Yeah.
Okay, thank you, sir. Here are some questions, sir.
Yeah.
Minimum investment work for entrepreneur in field of nutraceuticals.
So it actually depends on what you want to do. So whether you want to get into the product development, or the contract manufacturing, or the CRO services. So if it is CRO services, it will be very less. Probably you can start with 2 to 3 crores. And then if it is like R&D company, you need to have like at least like kind of 5 to 8 crores. But you have to continuously they get the investment. So 3 to 5 crores is just to start, but it will not be sufficient. So to tell you briefly about, like, so far we spent around like more than 25 crores in this company for developing all these products. So this is the amount of money. But there are like investors, once you have good products and good stuffs in the company, so they will come and invest you. So you need to like take money and then invest that for expanding your business. So that's how this business works.
Okay, sir, thank you. Now I request Dr. Pramod Esan sir to thank the speaker.
Dr. Somashekar sir, thank you so much for explaining your journey from the postgraduate to the national award-winning product development process by starting your own company. Probably it is going to be a very inspiring talk to the students. The students will experience, and then probably the tips that you are given during your talk is going to provide a lot of information to the students, whether for academics or to industry, or how to progress in the industry, what are the efforts they have to take into consideration. For probably it will be an interesting for some of the students who are financially sound can opt to go for an entrepreneurship in the later in the stage in their career. So with that, I'm going to thank you on behalf of the Department of Biochemistry, and as well as on behalf of the organizing team of this webinar for providing all the information, the journey from the whole perspectives. Thank you so much. With that, we want to thank you a lot. Thank you so much for being with us and sparing your time, sir.
Thank you very much. I'm really like honored to be part of this webinar.
Thank you, sir. Now I request Dr. Purnima Devi ma'am to introduce speaker of this session. That's Mr. VIJAYANANADA Bhatt, Manager, Food Division, ITC Limited, Bangalore, who is going to speak on topic opportunities for biochemistry in industry.
Okay.
I request Purnima ma'am to introduce the speaker, VIJAYANANADA Bhatt.
Sir, can I start and go ahead? I can introduce myself, no problem.
Sir, just a minute, sir.
Yeah.
Just within 2 minutes, we'll be starting.
Sure.
Problem with connections. So,
No problem.
We are getting one. Yeah, thank you. Hello, madam. What madam?
I request all the participants to wait for 2 minutes. There are some
ಹಲೋ
technical.
ಸರ್, ಇವಾಗ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ನೋಡಿ? ಬೇರೆ ಮೊಬೈಲ್ ಇಂದ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಕೇಳಿಸ್ತಾ ಇದೆಯಾ?
ಎಸ್ ಎಸ್ ಕೇಳಿಸ್ತಾ ಇದೆ.
ಮಾಡ್ತೀನಿ. ಅದನ್ನ ಅನ್ಮ್ಯೂಟ್ ಅಲ್ಲೇ ಇಡ್ಲಿಕ್ಕೆ ಹೇಳಿ, ಮ್ಯೂಟ್ ಮಾಡಬೇಡಿ ಅಂತ ಹೇಳಿ. ಅದೇ ಇನ್ನೊಂದು ಮೊಬೈಲ್ ಇಂದ ಮಾಡ್ತೀನಿ, ಅದೆರಡು ಹಾಗೆ ಅನ್ಮ್ಯೂಟ್ ಇಡಕ್ಕೆ ಹೇಳಿ ಸರ್, ಮ್ಯೂಟ್ ಮಾಡಬೇಡಿ ಅನ್ನಿ ಅಲ್ಲಿ. ಕೇಳಿಸ್ತಿದೆ ಅಲ್ವಾ ಸರ್? ಸ್ಟಾರ್ಟ್ ಮಾಡ್ಲಾ? Good afternoon, everybody. This is Dr. Purnima. It gives me immense pleasure to introduce Mr. Vijayananda BN. Mr. Vijayananda is Manager, Chromatography and Spectroscopy Lab, Foods Division, ITC R&D Bangalore. He obtained his MSc in Biochemistry from Kuvempu University Department of Biochemistry, PG Center, Davangere. Mr. Vijayananda was Research Assistant at Natural Remedies Private Limited, Bengaluru, from 2002 to 2004. He was Executive ASD at Strides Arco Lab, Bengaluru, from 2004 to 2006. He is empaneled NABL, EIC, APDA, and FSSAI auditor from Government of India. Till date, he has carried out more than 100 external non-ITC audits in individual capacity. He has qualified as a lead assessor. Mr. Vijayananda has 19 years of experience. He has functional expertise in handling quality assurance, activates in packaged food industry with key focus on instrumentation analysis. His major contribution is in establishing in-house capabilities for contaminant analysis with a main focus on pesticide residue analysis using LCMS-MS and GCMS-MS and heavy metal analysis using ICP, OES and ICP-MS. Mr. Vijayananda has been involved in establishing in-house capability for food authentication and food fraud using techniques like HRMS and ICP-MS methods. He has been associated with implementation of method development and validation protocols as per various guidelines such as SANTE, AOAC, ICH, etc. He has been associated with improving and maintaining quality systems by conducting audits in various plants of ITC across India. He has been serving as empaneled NABL, FSSAI, EIC assessor from 2016 and worked as TA in the field of residue contaminant analysis and other food analysis. He has been involved in statistical trend analysis for contaminants also. Mr. Vijayananda has been handling various research projects in the area of foods, analytical chemistry, and he is heading lab operations of GLP, compliance of lab, coordinating and leading instrument lab activities, providing instrumentation support for product development and QA activates, providing technical support for setting up of QQC lab across India, coordinating and monitoring all analysis as a part of FSSAI and other regulatory requirements, looking after all nutritional labeling analysis for product labeling, conducting audits of QC labs across India, and building new analytical research platforms like food authentication, food metabolomics, green chemistry, lab automation. Today, Mr. Vijayananda is amid us to online enlighten us on opportunities for biochemistry in industry. So now I request Mr. Vijayananda to take over. Thank you.
Thanks, madam. Thanks a lot for your kind introduction. So just give me a minute. I'll share my screen. Yeah. Is this screen visible?
Sir, visible.
Yes, sir. Yeah, thanks a lot. Thanks a lot. First of all, I would like to thank Kumar sir, Purnima madam, Gopal sir, so for giving me this opportunity. And it is a great feeling and honor to talk in the same college where you studied few years back, and coming and talking, giving a talk in the same campus and university. So I'm really feel privileged and to be part of this webinar. And so let me start with a brief introduction of myself. So I'm from Chikmagalur district, a small town called Ajjampura. So I have been done my all basic education there only. And from there, actually, I studied in Shumoga Sayadri College, my graduation. So then fortunate to get an opportunity to study in PG Center, Davangere. At that particular point of time, it was part of Kuvempu University. So where I was able to get guidance from Kumar sir, Purnima madam and Gopal sir. So there actually I started my career. So in 2002. So today I'm going to talk about my journey. So how it was there in the industry from past 19 to 20 years. So what are the opportunities and how one can choose to grow in the industry. So that's what is going to be. It is completely non-technical. You should pardon me for that. So I'm not going to talk any science today. Yeah. So when I came out of the college, so like most of you, I was also puzzled that actually what to do next. So because academics is very different and industry is completely different ballgame. And I was worried that actually what I'm going to really do. So is there any career for me? So is I'm going to reach somewhere in my career? So these are all the questions everybody will have. So I was also having similar doubts. So these ambiguities, I started my career. So because most of my family, so everyone who are there, so they were all actually techies. So software engineers, and they were having clear-cut goals. There were multiple companies to approach, and they were having lucrative careers. So they were on. So but actually coming from completely non-traditional degree, so which is biochemistry. So my father is a doctor. Actually, he always wanted me to become a doctor. But unfortunately, I was not able to get a seat in medical, and there was no interest in mathematics, made me come to this field. And after doing the course, I was not clear that what to do again. So then actually really, I started my career with a small company called Natural Remedies in 2002. So which was a small company at that particular point of time. Today it has become a little bigger. So it is a big company today. So I would just want to share so that how was my experience in the company, what are the opportunities, what are the lacunas, so where we should work, how we should focus. So my talk would be basically on that. So when I started my career in 2002, so the job was very challenging. So it was actually when I started, so there were not much options for biochemistry students. Today, you guys are really fortunate. There are multiple options. So that particular point of time, there was not much of an opportunity. So I started in Natural Remedies, which was a small company. The advantage of small companies, you can go to any lab and you can start learning. But the biggest drawback is salary. So you will not have big salary. So I started my career from past, say, a couple of thousands. Today, actually more than a couple of lakhs. So if you ask the journey, was it easy? No, it was not. Definitely not. But was it exciting? Was it actually lucrative? Was it enriching? Yes, of course it was. So the small company, I was telling you that actually the biggest drawback is the salary. So, but you can learn. So I learned actually first formative years of my career, I was focused only on learning. So whatever I was able to learn, I was learning. So from whoever I was able to learn, I was learning. So that made me actually landed up in a company called Strides Lab, which was a that particular point of time, it was around 5,000 crore company. So it was a mid-sector company. So wherein actually it was into pharma, it was into US FDA setup. So that was the first laboratory in Bangalore to undergo FDA and get FDA certificate for anti-cancer drugs. So I worked there for a couple of years. So there actually I learned many things, mainly the instrumentation techniques like HPLC, GC. So those techniques, and more than that, I was able to learn basically the documentation part, which is very important in pharma industry. I was able to learn that. So there, the biggest problem was actually there was no time. So people will be always occupied. There will be so much of job, and if one mistake of you can actually lead into big disaster, and it may even lead into the termination of the job. So you'll be always working under risk. So then slowly I moved. Actually, I thought actually it will be not wise to work here in this kind of environment for a long time, even though money was okay. I would say it was okay. But then I shifted to a company called ITC Limited, so which is one of the biggest FMCG company in India today. So it may be the third biggest company in India. So we're doing a turnover of more than $20 billion. So there, when I started the career, the advantage was it is a huge company. The brand name was good. The job security was wonderful. but biggest challenge is growth, because in bigger organization, the growth would be very, very slow. So, but then what I focused and what I learned there is actually you need to upgrade yourself constantly. So your skill set and your mindset. So what I mean by skill set is all learning all around you. So not just what you do, all around you, whatever possible opportunities to learn, everything you learn and upgrade your skills. So that can be learned. So what about mindset? Mindset is the champion mindset. So which is actually all the speakers today. So if you ask them, was it easy? It was never easy. So they have put their hard work. So they have put their actually, they never give up. They never give up. So just now actually Dr. So SOMASHEKARA was talking. So if you ask him it was easy, he will tell 100 things. So the journey would have been not very easy. So entrepreneur journey, he would have faced multiple challenges. But what took him there was probably the attitude, what he had. That is the mindset. So those two things actually someone can focus definitely, so he can reach. So is there an opportunity for biochemistry? So I would say yes. The good news is there are multiple opportunities today. So in the field of R&D, there are actually multiple career options, especially in drug discovery. So then preclinical, biopharmaceutical, project management, there are hundreds of opportunity for biochemistry students. When it comes to services sector, QC, QA, so R&D, there are actually multiple sectors again. So where there are multiple companies which are operating in India, abroad. So there are multiple opportunities. So if somebody wants to get into sales, yes, there is a lot of opportunities there also. Business development and other things. So even in the product development, yes, so there were multiple opportunities. So develop nutraceutical products, as SOMASHEKARA was telling. And then actually there are multiple opportunities in food industry to develop new products, and actually in biomedical industry, yes, there are multiple options for product development. And then in medical affairs, if you want, actually there are a lot of opportunities today available. So I'm going to briefly touch about few opportunities which are actually really booming today, and actually which can be a career option for you people. So generally the biochemistry, if you see as a major, so there are multiple opportunities as I was telling. So these are the few opportunities, especially the common ones and most explored ones are the professors, and then actually physician, biomedical engineer, these are the ones. Specialized will be in food sector and then medicinal sector. So then actually few people who wants to get into education sector, they will do B.Ed, and in few of my classmates have done that, they have become teachers. And then by biotech, pharmaceutical, sales representatives, there are actually hundreds of opportunities today for biochemistry. So what are the job profiles look like? So these are the few things which I have taken as an example to explain. So the most explored and actually in the industry, so people get into is into QC QA jobs. So what is QC? What is QA? If someone asks, so the answer is very simple. So QC is something which is routine. So every day you will be doing. So the checking the product quality and as well as the raw material product quality. So these are the things you will be doing. So this will be a function of generally manufacturing. So what is QA? So which is the function of management. So wherein you would be constantly checking how to improve the quality, and you will be assuring the quality, which is next level of quality control. So what are the skill set you need to develop to get into this particular job role? So it's mainly you need to learn. First thing is about proximate analysis. So which is all the ash, acid insoluble ash, moisture, these kind of analysis. The second thing is develop your skill sets in the area of mainly instrumentation analysis like HPLC, GC, other chromatographic techniques. So, and then spectroscopic techniques. So this is the requirement of this particular role. And then comes to research associate in basic research, FMCG, food pharma. So this is completely basic research. So wherein the skill set would be, so having more skill set, building towards, so the clinical assays, enzyme assays, and then actually the multiplet readers, ELISA, then all these PCR, these kind of techniques, wherein actually research in FMCG probably would be, say, all these claims what they make in FMCG product, like actually it lightens the skin tone. So this particular cream will give me more moisturizing capacity. So all these actually the claims is substantiated by lab studies, which are mainly done by biochemistry people. And in pharma nutraceuticals, yes, the label claims like anti-diabetic, the food is actually which can prevent you getting the diabetes, or it will, the low GI foods. So these are the claims which food industry makes. So these claims are substantiated by people who are from the biochemistry background. In pharma, there are actually multiple things. So already people have explained that. So you people have heard it. So that particular role is done by biochemistry people. So that skill set you need to build before getting into those jobs. And then clinical biochemistry is the most explored one. So clinical biochemistry people, generally they get into testing the patients and giving the reports to the doctors. So this will be mainly into diagnostics. This is a diagnostic industry. So someone who wants to get into that, so probably may have to build their skill set in clinical diagnostic sector. And then the pharmacovigilance. So this is a huge area which is coming up now after the entire world corona. So people have understood. So one thing that actually pharmaceutical industry is very important. The drugs are the one which is going to save the life, but whether the drug is safe. So the question is whether the drug is safe. To do that, so we need pharmacovigilance. So where actually all these clinical trials has been done, and the data management for clinical trials is the one which is huge. So the major sector here is pharmacovigilance. The skill set one need to have for pharmacovigilance is the knowledge about computers, so good knowledge about computers, I would say. And then it is completely a dry lab. The people who are ready to work in a dry lab, so can get into that. And they need to have a basic understanding of clinical studies. So what people do in clinical studies. So if one can develop the skill set in those, there are few courses also which are 2 months, 3 months, diploma courses and orientation courses, which are available today to get into pharmacovigilance. So then you can get into those, enroll into those courses and then get into these jobs. And for QC, QA, there are actually multiple trainings available. Even the Biocon has started now with training, which gives basic instrumentation technique training and ELISA, PCR, this kind of industrial training. So that even people can enroll into that, and then they can get into the QC, QA industry. And these are the top pharma and food companies in India. So you must have all heard about ITC, MTR, Haldiram's, so Azad, and then actually I must have missed few. So there is Nestle, so there is P&G, so HUL, and then top pharma companies, Biocon, Cipla, Glenmark. So the question is whether these companies take biochemists. Yes, indeed they take. So, but how to approach these companies? So better, first actually very important thing is, so have a reference. So get a reference. So there are multiple people from our universities and multiple people from our colleges are working already in these corporates. So get in touch with them, get a reference, and then apply. Or else actually apply through the portals. So which are, every company today has a portal. So it is easy to apply. But 90% of the chances you may not get a response only if you apply in the portal, because I have done more than probably 200, 300 recruitments now. So I never take any person without reference, because it is very easy if somebody refers and then the validation is already done for that candidate. So that's why references are so important today. And next, going into the pharmacovigilance. So these are the few companies which I was talking. The entire world now is moving towards India, because actually it seems that actually people are not having belief in China. So pharmacovigilance, before India, China was the biggest huge market, but today the pharmacovigilance industry has slowly moved into India, and there are giants like Covens, Equia, Novartis. So these companies, the startup salaries are also very good, and even the career prospect is very good. Another 10-15 years, the pharmacovigilance area is going to boom. So that's what actually the studies say. And there are multiple drugs which has to be launched, and every drug has to undergo clinical studies and clinical trials and clinical validation. So these are the industries to look for. And here I would just give you an insight that actually how you should choose a career. So first thing is each and individual. So first have very important thing is list down what is your goal, what is your talent and interest and the values. And second thing is select an occupation, 5 to 10 career options. So narrow down, there are multiple career options, just stick to 4 or 5. So that is enough, good enough. Why I am telling this is actually because otherwise you will be confused. And second thing is, so you need to develop, I told you that you need to have some skill set before getting into the industry. It would be very difficult to focus for more than 5 industries. And then your interest, whether this is my cup of tea, whether I will be able to see myself 10 years down the line in this particular career. That is very, very important. If I am not able to relate just because that they pays well, so if you get into such industry without your interest, so it would not be a great decision. Because in my career, I have faced that. So there are a couple of companies I worked, I didn't put in my portfolio. So I was not able to work not even more than 2 to 3 months. So that is not going to help us just because they pay more. So first, actually, the first and foremost is loving the job. So what I do. So then actually the hard work won't be hard work. It will be fun. So then explore the options, learn about educational or training requirements, job duties. So these things you need to discuss with your seniors. Promotion, opportunities. So you will not get it anywhere else. So there are a lot of people, I said, actually in the industry, you get in touch with them and actually check with them that how is this particular field. So how is the growth and what is the really the nature of the work. So then you will be able to correlate, okay, whether it will suit me or not. So then narrow down the list, eliminate everything else, reviewing one after the other, and then set a goal. So this is very, very important stage in the career. Why I'm telling this? Because without a goal, you are going to go nowhere. So now the Euro football is going on. All of you are aware, Euro Cup. So the best football players are playing. So those football players, if they are asked to play, they play fantastic, world-class football. Asked to play without a goal post, whether you are going to love that, whether they are going to love it. So without goal, if you start your career, that will also be, it will be in the similar fashion. So you're going to go nowhere. So that's why goal setting is very, very important in career. So have clear-cut, measurable goals. So then plan. So create a career option and actually then you are going to achieve it. So then obtain a training. So it may be professional training, it may be personal training. It is actually find out a mentor or enroll into some courses and self-ready. So is training or actually the learning is going to end just once after you join the career? So the answer is no. So you need to be constantly upgrading yourself in terms of skill set. So how, because, and why? So you have learned that. So actually it is very, very important that, so to get yourself upgraded after joining, ITC, I have probably done at least 10 to 15 courses in and around. So nobody has told me to do that. Only reason I did, because I was seeing myself. So as actually speaker, Jack Ma, so who is founder of Alibaba, he tells that a person's career, so it is actually always dependent not on what he does from 9 to 5:30. It depends on what he does after 5:30. So that's where this learning and skill set development will come into picture. And then, so how that career graph or growth in the industry looks like. So first, everybody will start from clerical job, or actually it will be say JRA, RA, or these kind of jobs. So where the basic objective of the job is to follow instructions. So is it something wrong in following the instructions? Not at all. Because a good leader is always a good follower. So first formative years of career, it is only to follow the instructions. So then only we can become better and reach to the next level, where I will be a completely independent worker. So there my responsibilities would be doing my job without any supervision. And then you will be elevated to the supervisor position. So where you will be handling a small team. And your job would be not only doing your bench work, you will be always, you will be assisting other people also around you. And then comes the managerial role, where you will be start becoming a leader. And then you will move in the ladder, like actually strategic, where actually you will be defining the strategies. And actually you will be part of management. So this is what is the career growth looks like. And then, so how to approach for these job companies? So there are multiple options today. You people are really lucky. So I was telling that networking, so it is very, very important. So to get into career and also grow in the career. So because networking is network of yours is always net wealth. So that's what my mentor is always used to say me. So these are some of the sites where you can get, so you can search people who are in the same industry, who are in the same job profiles, what you're looking for, people from same background, qualification, and a lot of your academic seniors who are from the same colleges. You can get touch into with them and then ask for their help. They would be happy to help. Most of them would be happy to help. So it is just that I actually approach them and then I talk to them. So I get mentoring from them. So this is very, very important today. So otherwise it is very difficult to get into the good job. And also it is not, you will be not able to build your career in a progressive way. If I work in silos, if I am not networking. So these are the platforms where you can network and then you can actually build your career. So any questions, I would be happy to take. Otherwise, I am done, more or less I am done. Thank you very much.
Thank you, sir. There are few questions regarding this talk, sir.
Sure.
Basic science students will not have any knowledge of marketing. How to manage to get into companies like food and pharmaceuticals?
Can you repeat the question?
Okay, sir, I will. Basic science students will not have any knowledge of marketing. How to manage to get into companies like food or pharmaceuticals?
So you want to get into marketing career? I was not still I am not clear with the question. Do you want to get into marketing, or the question is how to get into industry?
So your question is how to get into the industries like food and pharmaceuticals without the knowledge of marketing.
So you don't need, first of all, you don't need to have a skill in marketing to get into industry, because this is completely technical domain. So where your skill set should be, as I told you, if you want to get into QC, QA job, so your knowledge or skill set should be towards learning the proximate analysis, instrumentation analysis, and other stuff. And if you want to get into a career where you want to get into research, it would be more on other techniques which I told you. So which are basically ELISA, PCR, and Western blotting, those kind of techniques. So, and if you want to really get into say food industry, what I recommend is, so better you just read about few regulations or like say guidelines, so which are freely available. So like FSSA is a guidelines where actually every food need to comply with this regulation if it has to be sold in India. So FSSAI is one guidelines. So there are actually multiple books available, multiple actually now online courses available. So which actually will help you to gain a knowledge about food industry. And also, so in pharmaceutical industry, if you want to get into, so actually get your skill set aligned towards HPLC, GC, so these instrumentation techniques, which is already part of your curriculum. So only thing is you must have not been exposed to these equipmental techniques. So you can, in YouTube, today actually everything you can get in YouTube. So you can get online courses, classes there, actually courses are available. So there are multiple videos which explains about these techniques. So you can gain knowledge about them before attending the interviews. And if you ask when I should start that, so you should start, this is the right time. When you are there in the college, that time only you should actually have a goal that I will land up in this industry and start building your skill set. That would be the ideal scenario.
Okay, sir, thank you. Here, another question from the participant. Can we get jobs in food, pharma, forensic by just finishing MSc in biochemistry?
Definitely you can get.
Okay, sir. Sir,
Yeah.
here another question, sir.
But you need to have the,
okay, sir.
you need to be very clear with your basics. One is your foundations, and then some additional skill set. Probably you can just acquire by reading.
Okay. Sir, here is another question from another participant. After my PG, how to choose either pharmaceutical industry or food industry? Please guide me, sir.
See, fortunately, both food and pharma, both industries are recession proof, 100% recession proof. So whether Corona comes, whether actually financial crisis comes, so these, both the industries are going to be there because people has to be healthy, people has to eat. So that's why these are, both the industries are 100% recession proof. So these industries, where you want to go, it's all depends on your interest. So actually there only I told that goal setting is very, very important. So you need to understand that actually you need to talk to a couple of people. So actually you should have an understanding whether pharma industry, if I have to go. So you will be actually doing something which is related to pharma, where actually I would say the job is more or less monotonous. So you will not have actually flexibility to do much of, say, research and development activity because all the molecules, so only in drug development you have a scope of doing new work. Otherwise it is mostly in pharma QA QC, you will be end up doing the same work, same job. If you are okay with that, so then definitely pharma is an avenue. In the food industry, it is evolving. So you have a lot of opportunity for learning and getting into new developments. So that depends on individual's career interest. But both the industries are equally good. You can get into both the industries, and the learning and earning both are good.
Okay, so thank you. So here is another question by the participants. Is dry lab have more scope or career than that of wet lab? Please let me clarify, sir.
I would say actually you cannot quantify like that because it depends on the individual capacity, individual interest. So a person who is excellent, having actually a lot of interest, who just love his job and ready to do hard work, can grow in any of these two. So a person can grow in dry lab, a person can grow in wet lab. It depends on individual's interest and actually, but if you see the career options today, probably I would say, so it is 50-50. So both the industries has job opportunities. But why I was actually focusing on pharmacovigilance, because that is booming up now. Actually, it is just started and it is coming up very well in India. So where actually you will find more and more opportunities for next 10-15 years. So whereas food and pharma industries are more or less saturated today. But if you ask me, a person like me cannot work in dry lab. I cannot sit and do that work. So that again depends on the career, that individual career interest.
Okay. One more last question, sir. Those who passed out in this pandemic and who have no research experience apart from our PG projects, in order to gain research experience or learn any techniques for which we need the practical knowledge. But sir, apart from reading research paper, how a person oriented towards research can use this lockdown period?
Actually, I would say this lockdown period, it is actually a blessing for people who want to really develop themselves, so their skill sets, because actually today internet has everything. So online has everything. So there are multiple courses which are available online if you search, and there are multiple seminars, webinars which are happening like this every day. So if one is keen on learning, they can learn actually a lot of things, because you don't have to really go into the college. So you you are not actually, you guys have more time, I would say, so than actually what we used to have. So, but coming to the experience in research, so hands-on experience in research, so that probably you may get only once you get into the lab. So there definitely, yes, you people have to do some additional hard work once after getting into the industry, because everybody understands today that you may not have such exposure, because so if you are not, actually the labs are not open every day, probably there is little bit of, yes, so you guys have a little bit of disadvantage there. But otherwise, in terms of gaining knowledge, in terms of having understanding, I would say that you people are more blessed.
Okay, sir. Thank you, sir. Thanks a lot. Your words means a lot for our young minds. Now I request Purnima ma'am to thank the speaker.
Thank you.
Thank you, Mr. Vijayananda, for your wonderful lecture on opportunities for biochemistry in industry. It was really motivating and helpful for our students especially. Thank you once again for your valuable time. Thank you.
Thanks a lot, madam. Thank you. Thank you.
Okay, now it's time for validation. I request Dr. Pramod Essen sir to deliver the vote of thanks to the participants and gathering.
Honorable Vice Chancellor, our respected Registrar, Register Evaluation, faculty members of Department of Biochemistry, research scholars, students, and all the participants across the nation. Very good evening. Myself, Dr. Pramod, Associate Professor, Department of Biochemistry. It is indeed a great pleasure for me to propose vote of thanks at the end of the concluding session of this one-day national webinar on frontiers in biochemical sciences, organized by Department of Biochemistry, Downstream University, in association with Society of Biological Chemists India, Downstream chapter. First and foremost, we would like to express our sincere thanks to our Honorable Vice Chancellor, Professor Sharanappa V. Halsesar, for his encouragement and constant support in organizing this webinar. I also take this opportunity on behalf of the organizing team to thank our Registrar, Professor Gayatri Devraj Madam, and Register Evaluation, Professor Anita Madam, for their support and encouragement in executing this webinar. I take this opportunity to thank all the 6 member research persons who agreed to share their research experience and work and their career and as well as experience with our students and participants during this webinar session. First, I'll take up the research person, PANNAGA, who delivered the keynote address on photosynthetic cell factories, the 4th dimension in industrial biotechnology. Thank you, sir, being a research person and then delivering your excellent talk to enlighten our student. Then I also thank Dr. SHIVAKUMAR, KEERTHIKUMAR from Peter MacCallum Cancer Center, Melbourne, Australia, who spoke on current trends in bioinformatics and introduction to sequencing techniques. Thank you very much, sir. Then, I also take this opportunity to thank Dr. VENKATA, DK, CSR, CNAP Research Center, Bangalore, who spoke on role of lipids in signaling mechanism. Thank you, sir. Then, on the behalf of department, I also thank Dr. VENKATESHWAR Reddy, biologically related, Hyderabad, and who delivered a talk on downstream processing of vaccine candidates. Thank you, sir. Then Dr. SOMASHEKARA's experience with reference to the nutraceutical industry, and his talk was very, I thank him on behalf of the Biochemistry department. Then finally, I thank Mr. VIJAYANANADA, who is in ITC Limited Bangalore, who shared and delivered a talk on opportunity for biochemistry in the industry. Thank you for being with us and enlightening our students and motivating to choose a career options after graduating from biochemistry. And I thank all the research persons for sparing their valuable time with us and giving their wonderful ideas and experiences to the Department of Biochemistry. Now, I take the opportunity to thank Dr. VENKATESHWAR sir, Chairman of the Department of Biochemistry, Dean, Faculty of Sciences, Downgrade University, and also the force and the team leader for planning and executing of the present one-day webinar. Thank you, sir. And I also thank our senior Professor Gopal Advita sir for constant feedback and engagement in organizing this webinar. And our heartfelt thanks to our colleagues, Dr. Santosh Kumar sir, Dr. Purnima madam, Ms. Ranjita, Ms. Shalini, Dr. Pratap, Mr. Shekhar for their valuable support and involvement in organizing this webinar. I also thank all the research scholars and the students of the department for involving and benefiting from this national webinar. And a special thanks to Shalini for presenting the invocation for mastering the whole systems during the webinar. And finally, I thank all the participants across the nation for actually participating in this webinar. Thank you all who have directly or indirectly contributed for the success of this one-day national seminar on frontiers in biochemical sciences. So on behalf of organizing team and Department of Biochemistry Downgrade, I once again thank you everyone. Thank you all. Have a great day.
Thank you, sir. I request all the participants to fill the feedback form by clicking on the link which is provided in the chat box in Zoom platform and in the comment box in YouTube. And this link is also shared in the Telegram groups. Please fill the feedback form by clicking on the link. For those participants who will successfully fill the feedback form, for those only, e-certificates are going to be sent through your registered mails within 3 days. So with this, we are now at the end of the today's webinar session, that is Frontiers in Biochemical Sciences. On behalf of our Biochemistry department, I thank all the dignitaries, resource persons, and all the participants for making this webinar session very successful. And finally, I thank all of you for giving me this opportunity. Thank you all. With this, the webinar is going to be end off.
Okay, thank you very much.